Everyone. Okay, now recording to progress. Good morning, everyone. I'm going to talk just a brief update on what we've been looking at with the influence of forest management on above ground carbon. And oh, come on. Great, now it doesn't want to advance. There we go. So one of the things I did want to do is I want to give a big thank you to all the collaborators in this study, Regional Water Authority, the Nature Conservancy, White Memorial Foundation, also with some funding and help from the Forest Service, the Forest Ecosystem Monitoring Cooperative, and the Forest Stewards Guild. So certainly, I think we're all aware now that there's a lot of concern about the changing climate, and things have been observed to be getting warmer, but also it's been observed getting wetter. What's going to happen in the future is going to be kind of interesting. And one of the possible solutions people have had, or a part of the solutions, has been to have forest as a sink for carbon. When we look at the amount of carbon that's out in the woods, we find that roughly half of it, and the part which is most volatile, is the above ground biomass. That's what's in the leaves, the trunks, the leaves, and everything else. Roughly half of all the carbon is in the soil, and that is remarkably stable so long as you have forest or trees growing on it. What does happen when you harvest trees, though, is a lot of the carbon is put into durable wood products, and that's a long-term storage solution. So just to do some explanations for those of you who aren't aware, there's a difference between, when we talk about carbon, between sequestration and storage. So sequestration is we're adding new carbon into the system. So that's carbon taken out of the atmosphere and which is put into storage, usually in woody structures. Storage is the amount of carbon which can actually be found on the site. So that's something you can go out and physically measure which is out there. But then you have harvesting, and that is going to reduce the amount that's stored in forest, but it generally, after a 5 to maybe 10-year period, lag period, is going to increase sequestration. But when you do harvesting, you also provide durable products. I want to give a big thanks to some of us in my generation. Remember George Stevens? George set these study plots up back in the early 1980s, not to study carbon, but to study the impact of different cutting practices, and I'll mention all those, on forest regeneration. But one thing, if you knew George was very meticulous, and he took incredible amount of information, and that allowed us to use this study to actually look at what was the impact of different cutting practices on above ground carbon. So here's an aerial shot of one of the study areas. This is in Beaver Pond and White Memorial property. And there were 6 treatments. There was an uncut control. There was a 16-inch diameter limit harvest. One of the things too with the diameter limit harvest when they went in there is George, and when we did it subsequently, we cut down all UGGs, unacceptable growing stock trees which had a lot of decay. And that would have an influence also on carbon, because if a tree is decaying from the inside out, even if it's growing on the outside, we're losing carbon on the inside. We had a shelterwood where they put in the initial shelterwood cut back in the early 1980s. And in 2000, 2001, had complete overstory removal. We had multi-age crop tree management. Originally, George called that coppice with standards. The idea there is long-term is to develop 5 different age classes. And every 20 years, we'd come and cut the largest trees in the stand. So roughly 10, 15 trees per acre of the largest trees were cut. The remaining trees were left. You picked around 10 to 20 new trees per acre, which the goal would be in 80 to 100 years. Those would become the harvested trees. And all other poles were cut at the same time to give plenty of growing space for the new crop trees. Also put in a silvicultural clear cut. It was originally described as a 2-inch diameter clear cut, But from the photos, by God, I think they cut everything down to probably 4 or 5 feet high, just because the operation was so intense. Then they had a 12-inch high grade. And that was a commercial, we called it a commercial clear cut or a high grade. Everything of commercial value that was 12 inches and greater was harvested. And I'll talk a little bit more about that in a second. So if you just want to see what they look like, sort of a nice little profile view, that's what they all looked like. Unmanaged forest preserve, multi-age crop tree management. Think of multi-age crop tree management, it's sort of like a city park where you've got 5 different age classes. And it makes for a pretty interesting area because you get a lot of shrubs coming in. And surprisingly, we found we got a fair amount of oak regeneration, although I'm not going to talk about that on now. The 16-inch diameter limit cut, shelter wood, a high grade or commercial clear cut, and the silvicultural clear cut. Want to give a thanks to a lot of people who've worked on this over the years. There's JP using a prism to do some measurements. Alexa Mora, who helped us with measurements one year. There's David out there marking a tree. There's Lucas helping us get some cookies to age the trees. And Johnny Yenzo's down there doing some of the regeneration counts. One of the exciting things going forward is we're now working with NRCS to look at the impact of these different cutting methods are going to have on below ground soil carbon. Remember I said below ground is about 50% of the carbon? Well, with the data we've taken in the past, we haven't had a good understanding of what's going on down there. So Don and Milton and others have been helping us over the years, and there's a good chance a new scientist at the experiment station might be helping us going forward on looking at how different management affects below ground soil carbon. So basal area, this is just one of the things, you know, as foresters, we take a quick look at and it's like, oh my God, look at all these zigzag lines. So I'm going to have similar graphs up a few times. Let me just make sure I have my clock here so I stay on schedule so Vicki doesn't yell at me. So on the x-axis on the bottom, we have the timeline. Everything started back in early 1980s. We had the harvest. We allowed things to grow. In 2000, 2001, we harvested again. Let things grow. And what doesn't show on here, but I will show in subsequent graphs, is we had the third round, the third cutting cycle was completed during this past year, and there should be wrapping up within a month over at North Madison, which is the third cutting area. So if you look first at the black line with the little gray circles up on top, that's the uncut area. And what's interesting here, I'll talk more about it later too, is the basal area was growing slowly on these stands, but has since plateaued out over the past 20 years. If you look at all the other ones, you can tell depending upon the cutting intensity during the first cutting cycle, they all dropped. But look at the lines between early 1980s to 2000. You can see that there was pretty rapid basal area accumulation on all these plots. And one of the things I think is kind of interesting is if you look at the blue line with the triangles, that was the silvicultural clear cut. And the silvicultural clear cut now, approximately 40 years after the initial clear cut, is now back up to almost the same basal area that it had initially. And if you look at the green line with green triangles, that was the shelter wood. The shelter wood is also following the same trajectory of increasing basal area. Okay, as foresters, as Judd White used to like to say, we're eco-brutalist timber beasts, so we worry a little bit about saw timber volume. And you've got to pay to have operations done in the woods. If you're creating habitat, if you're improving forest stand health, whatever. And I'm just going to look here to keep it a little bit simple. I'm just going to talk about oak volume changes because while there was a little bit of sugar maple in these stands, the real economic driver in these systems was oak. And if you look again at the black line up on top, oak volume actually peaked 20 years ago in these stands. We've had some disturbance on those, and that's how I'm going to wrap up the talk, talking about the influence of disturbance, especially on carbon. But Oak volume actually peaked, like I said, 20 years ago, and these stands were about 110 years old. If we look at the purple line with the diamonds on it, that was the multi-age crop tree management. And you can see we've had a pretty steady sawtooth. And it looks like that's actually a way to maintain oak out in a stand, which is kind of exciting because we really don't have a way to previously to maintain large oak in a stand while we're having commercial harvest. And I should say all of these were commercial harvest. If you look at the triangles, the blue and green triangles at the very bottom, that was the clear cut and the shelterwood plots. Those trees have not grown large enough yet to really have any appreciable commercial value. If you look at the orange dashed line with the orange squares, that was the high grade. Boy, you can tell it was brought down to nothing back in the initial harvest. We did not have a second harvest on there back in 2000. Why? Commercial volumes were way too low. If you look on here, there's only about 1 MBF of oak board feet per acre, and that's just not enough to sustain a commercial harvest. There was a one this time, even though volumes were pretty low, so the recent ones. If you look at the diameter limit, the 16-inch diameter limit cut, and I probably went through the slide a little bit too much earlier. So oak volume was starting to increase a little bit because there was some residual oaks less than 16 inches. But this second harvest, we had to bring the diameter limit down to 12 inches to make it commercially viable. So sorry, 14 inches. And this last one, we had to bring it down to 12 inches to make them commercially viable. And what's interesting with all the cutting methods, except for multi-age crop tree management, after 3 cutting cycles, essentially there's no oak left on the site of any commercial size. One of the things you might be looking at is, well, what's the cumulative economic impact of all these different cutting methods? And the yellow bar at the beginning is, hey, that's what the initial value was when we went onto those stands. And I gave stumpage prices of 150 MBF dollars per for 1,000 board feet of oak and $50 per 1,000 board feet of others, and estimated the economic value. What's interesting is you can change those numbers, and it's relatively insensitive because we're looking at percentage values here. And the surprise is, except for multi-age crop tree management, that's the MACT, pretty much they all had the same economic value over the last 40 years, over 3 cutting cycles. Multi-age crop tree management, because we're maintaining oaks on the site and because we're continuing to grow oaks on the site, actually has the highest economic value. If anyone out there who's an economist, no, I did not look at discount rates and all that. This is just raw gross numbers of dollars coming off. But this talk is supposed to be about altered carbon. So how does carbon change in the forest over time? This graph here, I'm just looking at carbon relative to the initial amount on there, because I think a lot of our eyes glaze over when we start talking about, megatons per hectare or megatons per acre out there. So I just said, okay, what's the initial value out there and how's it changed over time? When you look at the uncut controls, we've actually seen that for the first 20 years, between the 80s and 2000, there's a pretty steady increase in carbon. But for the last 20 years, there's been a nominal increase in above ground carbon over these 3 areas. When we look at the other treatments, again, you're going to see that zigzag pattern. But let's look at a little bit more closely at a couple of them. So if we look at the blue line again, the blue line with the triangles, that was a silvicultural clear cut. What I think is pretty amazing is that 40 years after that clear cut, we're already up to 80% of what the initial above ground carbon was on that site. And if you were to project that out in another 30 years, it's going to have the same amount of above ground carbon as we had on the unmanaged stands, which is really, really exciting. If you look at the shelterwood, we're seeing a pretty similar pattern to what we saw on the clear cut stands, that we're seeing rapid carbon accumulation over time. we look at multi-age crop tree management, that's where it gets busy in the middle of the graph. That's the purple line with the diamonds. We can see that we brought it down actually to about 40% with the first harvest, and then increased up to almost 90% before the second harvest, dropped it back to 50%. Then it went back up almost to 90% again, and we dropped it back down again. So we're actually able to extract a commercial forest product out of the land, which are then put into durable goods. And we're able to see that those stands are able to rapidly recover. One thing, it's either good or bad, depends on how you look at it. If we look at high grade, that's the orange dashed line with the orange squares. It's the longer dashes. It actually, because we did not have a second harvest on that back in 2000, actually recovered by 40 years after the initial harvest, so that it had more carbon on the site when the stand was initially was harvested. Another way to look at it's a little bit, maybe a little less confusing in all those lines, and I'm going to leave it on here, is let's look at storage. This is the amount of carbon which is in live trees above ground over the past 40 years. And each one of these, you look in the bottom, it says pre and post one, that's before and after the first cutting cycle. Pre and post two, that's before and after the second cutting cycle. And again, pre and post three, before and after the third cutting cycle. So again, you can see in the uncut, it really had a pretty good increase for the first 20 years, but the last 20 years have not seen much of an increase. Silvicultural clear cut, we've seen that steady increase. And like I said, we're soon going to be at the level to the initial level of above ground carbon. Within another 30, maybe 40 years, we should be at about the same level as the unmanaged forest. Shelterwood followed a similar pattern. If we look from post two, which is after when the overstory was removed, we've seen pretty rapid carbon accumulation. And I should note that these include estimates of carbon which are in durable wood products, discounting for the fact that durable wood products gradually either go to an incinerator or to a landfill, because unless it's in a house, products have a given life cycle. And again, you can see that if we look for the high grade, we saw a fairly rapid recovery because we didn't have any harvest in that second cutting cycle. But now we've dropped it down. What's interesting, if you look in the lower right for the multi-age crop tree management, we've really seen that we're able to maintain a pretty good level and a good recovery of carbon throughout the entire three cutting cycles. So if we look at carbon sequestration rates, so remember, storage is what's actually you can find out there. And sequestration is the amount of new carbon which is being stored. And for the period after the first cutting cycle, for the first 20 years, carbon sequestration was higher on most of the treatments than it was for the uncut. And I'll explain why in a second. The second cutting cycle, I'll go even more detail on that. The amount of carbon that was being sequestered on those uncuts absolutely collapsed. So the question is why? Well, if we look at the diameter growth of these trees and we compare the uncut with the diameter limit in high grade, didn't include silvicultural clear cut and the Shelterwood and the MAC because I just wanted to have one symbol graph, but they're pretty similar, is we can see that where you had harvest out there, those trees are released. They have much more growing space. They have more light available. They have more access to soil moisture, soil nutrients. So the growth on those trees was much, much faster where we had a harvest. So the residual trees that were left were able to grow much more rapidly. This is all, you know what, I'm gonna skip that, it's a little too confusing. So if we look at forest management and carbon sequestration, one of the things that people have talked about is that more complex forests sequester more carbon each year. And more complex forest means you've got large trees and small trees and they all have access to light. But that unmanaged forests generally have more stored carbon. Well, here's one of the problems with unmanaged stands is when people like doing projections of unmanaged stands, they sort of talk about like there's never going to be a disturbance out there. They project them out as if there wasn't a disturbance. And on our unmanaged stands, we had a couple of disturbances. Over in the Bridgewater site, that was hit pretty hard by a derecho. And what's interesting is the damage was much greater in the unmanaged stand than it was to the adjacent stands, which had younger, smaller trees, which I believe were more wind firm because they were exposed to wind and they didn't have the protection of their neighbors. If you've ever seen where a derecho hit one or two trees go down, you start getting that cascading effect and have more trees go down because they lost the wind protection of their neighbors. The other disturbance event we had out there was we had multiple years of LDD. That's the new name for what we used to call a gypsy moth. It's now, I can never pronounce it, but anyways. So we had 3 years in a row of LDD defoliation on the North Madison stand. And we actually saw a big decrease in the amount of live above ground carbon that was stored on those stands. So if you want any more information, one thing I would suggest is a great introduction to this for those of you who aren't foresters is look up Forest Carbon by Paul Catanzaro and Tony D'Amato. It's a great introduction. It's in common English you can understand. And also Yankee Division Society of American Foresters has put together, I think, an outstanding trifold, which also explains that when we're managing forest, we're not managing just for carbon, but we're also managing for recreation, we're managing for habitat, we're managing for clean water, and I know the folks in the water companies know that. We're also managing, let's be blunt, for timber products, because one of the things we want to do is use products which are more carbon neutral, and wood is much more carbon neutral than are the alternatives of steel, cement, and plastic. So the take-home messages with this unmanaged forest, we've seen minimum change in carbon storage and board foot volume over the past 40 years because you know what? Disturbance happens. And in managed forests, we saw rapid increases in carbon sequestration and recovery of carbon storage because when trees are given more space to grow, they grow faster. So with that, I'm going to stop sharing. And Vicki, I don't know if I'm allowed to take any questions. I think I have a couple minutes.
Yeah, Jeff, that was fantastic. Looks like you have time for a few questions. I just looked at the chat box and nobody put any questions in the chat. So if you have a question, go ahead and unmute yourself and ask away.
Hey, Jeff.
Yeah.
Yes, David Irvin. Are you confident in the size of the study areas for each of those types of cuts? Because it almost looked like you would get a lot of shading or sheltered influence from the adjacent treatment areas. What was the size of each treatment area?
And David, that's a great question. The treatment areas were 5 to 7 acres in size, and you are going to get some spillover, some side effects. But I think that's pretty minimal. If you look at how different every one of the treatments were, especially with carbon and basal area, there was real clear-cut differences. I mean, significant differences. I didn't want to get into the stats on this. But I think, like I said, 5 to 7 acre plots, these aren't little Mickey Mouse plots out there. And I should say there was one prison plot. I don't want to get into details on that per acre on these plots.
Okay, thanks a lot, Jeff.
Sure.
Any other questions or comments for Jeff?
Hi, Jeff. This is Amanda from UConn.
Yeah. Hi, Amanda.
Hi. I was just wondering if you could talk a little bit about how you calculated the carbon on there. Are you just measuring mostly DBH and size of trees, or is there something else you're measuring to calculate that?
Sure. I used, let me pull it out. I use the above ground bioestimates from Chignacki et al. In 2004, which is primarily DBH. For the soil, I mean, for the harvested wood products, we actually figured out the volume of each tree because we took saw timber. We then converted that to merchantable volume, and then we converted it to there's a formula in Smith et al. 2006 of 0.765 megagrams of carbon per 1,000 board feet. But it varies a little bit depending upon what species you're using, but that's the approximate. And we also discounted because JP worked in FIA and has been doing this a long time. JP looked at call. So we took board foot call percentages and we discounted those from merchantable volume and also from carbon.
Oh, okay, thanks.
I'm just happy to see that somebody is looking at the carbon in the soil as well. I think that that's an important component, given that a tremendous amount of the biomass in a forest is actually in the soil. When you consider everything from fungi and bacteria to insect larvae to little tiny plants. So I think that's a very important consideration there. And there's a comment from JP in the chat.
Sure, I'll take a bottle of Pappy bourbon. Somebody's got $1,000. Oh. Well,
Anyway,
go ahead.
Oh, go ahead, Jeff.
No, I was just going to say thanks everyone for their time and hope to see you all on the other side of retirement.
When is your official date, Jeff? I've forgotten already.
84 days.
84 days. Cool. Okay. All right. Our next speaker on the agenda is Amanda Bunce at UConn. And I was just looking to see if you're there, and then you asked your question. So I know you're there. So whenever you're ready, you can start screen sharing and take it away.
Okay. Let's see here. Does that look like a PowerPoint presentation?
It's getting close. Yeah.
Okay.
You just need to put it in the slide.
Let's see. How about that?
That does it.
All right. Okay, so my name's Amanda Bunce. I'm from UConn, and I'm going to talk about our adaptive silviculture project. I talked about it at this venue last year, I think. So this is just an update of what we're doing. And I want to start off by thanking everybody that's involved. We have a lot of researchers here at UConn, both ecologists and forest scientists and social scientists working on this project, and a lot of graduate and undergraduate helpers. We have Maria and Courtney are our ASK project advisors. ASK is the Adaptive Silviculture for Climate Change Network, and they come from USDA, NIAX, and the University of Colorado. And then we have our Connecticut Deep people, and Christopher Riley over at URI is working on this project as well. So we got a lot of hands in this one. Okay. So my slide changed, right? Anyone confirm that for me?
Yeah, it looks good.
Okay, good. All right. So the impetus behind this project is the fact that there are changes to the climate that are happening right now, as Jeff described, as everyone probably will. And those changes are impacting the forest's ecosystems more rapidly than forests can adapt. So adaptive silviculture is one way that we can grow trees to help our forests be more resilient to these climate change impacts. These impacts include a multitude of forest health issues that may or may not have been there without climate change, but climate change is definitely exacerbating them and causing cascading effects and combination effects of these forest health impacts that our forests are just not used to. So adaptive silvicultural practices are whatever we can do to grow the trees to help them deal with this stuff. Adaptive practices are not especially widely used in Southern New England, but we have a lot of forest cover and we have a lot of human population that depends on that forest here. We have a multitude of benefits that the people know they're getting from the forest. Awareness of the impacts of climate change seems to be pretty prevalent among not only forestry professionals, but among people that use the forest users in this area as well, and landowners and land managers. Willingness among land managers and landowners to adapt the forest to climate change is pretty high, but adaptive silviculture isn't especially being used here a lot. So this study is our look into why and possibly how we might get it going a little more. So we look at the exurban forest as a socio-ecological system. That's a system that's influenced by both the social and the ecological conditions around it. And the forest is going to influence those things in turn. So as we use the forest and we manage the forest, so does the forest impact us. Our research questions are both social and ecological, and they include what are some of the considerations that are important for forestry decision-making on the exurban landscape. So what do we think about when we dive into forest management here? What are some feasible and effective silvicultural strategies to help get our forests adapted to climate change? And what kind of priorities, what kind of knowledges do managers of exurban forests have when it comes to these adaptive management strategies that we're talking about? So a lot of questions on this project. And to start answering them, we partnered with the Adaptive Silviculture for Climate Change Network. So this is a network of silvicultural sites around the US and Canada, and they are testing adaptive forest management actions. So the treatments, the experimental forest management treatments for the ask sites get co-developed in a workshop by local land managers, forestry scientists, and other local stakeholders. So people get together to come up with the treatment plans, the adaptive silviculture treatments that they're going to test out. There are 3 options, but you come up with a plan for all 3 of them. So you come up with something to help your forest resist the changes, something that makes it more resilient to the changes, and a way to transition your forest to be better adapted for expected conditions in the future. These sites in the Ask Network are generally 4 replicates of 4 treatments that are all 25 acres each. So these are some pretty huge experimental forest plots out there. Now, we're not going to find that kind of area in southern New England at all. So our exurban sites are not going to be exactly the same. They're going to be about 5 to 7 acres. We're going to do 4 treatments still. So that the 4 treatments is resistance, resilience, transition, and a control site that we don't touch. So 4 treatments of 5 to 7 acres each, and we're going to replicate them in different places. So they're not all going to be on the same forest. They're going to be in the same types of forest, but not right next to each other. So we did start out with a workshop in October of 2020. Of course, COVID hit, so it was going to be impossible to do it in person. So we did it in a virtual format. We created this really great story map, so our virtual participants could check it out online and look at soil maps and hydrology maps and all kinds of maps of stuff of the Mohegan State Forest in Connecticut, in Scotland, Connecticut. So the virtual participants were able to check those out. They were able to look at our tree species and volume measurements all over the site. And together, we worked with those people to sort of co-develop our 3 treatment plans to adapt the forest in that resistance, resilience, and transition way, but also to meet local objectives and encourage ecosystem adaptation, considering the local conditions. We did a site visit too, so those of us that were local went over to the site and broadcast our meeting in the woods to the rest of the participants online. Yeah, so Mohegan State Forest in Scotland, Connecticut. During that workshop, we were able to ask a couple of social science questions as well. So we wanted to know what people were seeing, what kind of considerations they're seeing with climate change when it comes to their forest management decision making. So there was a lot of uncertainty came out of that. People were very concerned about what they don't know. It's hard to make a forest management treatment plan when you're not sure what the future is going to bring. And that's not so much a question of not understanding what climate change does to a forest. It's the fact that climate change brings with it uncertainty. So we're not sure, we're sure they're going to be impacts, but we're not sure how bad they're going to be or what they're going to be. So that's why we want to foster adaptation, foster the ability for the forest to adapt itself as these changes come onto it. Another thing we looked at was people's objectives in their professional responsibilities, so as forest managers, and mitigating pests and diseases, promoting biological diversity, protecting water quality, protecting land from development, and habitat were all right on the top of that list as the most important things that were showing up in people's forest and land management jobs. Things that were not so important were improving hunting activities, privacy, fishing, firewood and timber production were down towards the bottom of the list. So those were objectives that are not super prevalent in Southern New England lately. So that was helping to inform our objectives as we hash them out for this site. All right, so these are the treatments that we came up with. We have one, you can see in the stocking guide on the side there, our trees were in the fully to overstocked area. We had been dealing with a lot of die-off from gypsy moth drought combination, 2016, 17, and 18, killed a lot of oak trees in the area. So we also looked at the stocking with that mortality removed. It was still in the fully stocked area. So then we created our 3 experimental treatment plans. And one was to resist the impacts of climate change. So we wanted to maintain that oak hickory dominance, promote the health and adaptability of the growing stock. And to do that, we were going to do a shelter wood cut. So a thinning now with an overstory removal 20 years from now to regenerate oak and hickory in that area. But in that area, we also wanted to protect reserves that had unique habitat, sensitive species, and there's a hiking trail that goes through that area as well. So our resilience site, we're going to bring the stocking down a little more. We want to promote conditions that really allow for the development of a multi-cohort of oak and hickory in that forest. So we're going to do patch cuts, potentially a controlled burn to encourage that multi-age regeneration, and possibly some planting of blight-resistant American chestnuts in that area. And our transition treatment, we're going to bring the stocking way down because we want this forest to sort of turn over into something that's going to be more effective in the future at handling what's coming, handling the conditions of the future. So we want to promote the stand development with our future adapted species there. So we want our multi-age structure. And we're definitely going to do some planting there of maybe not just chestnut, but also southern origin oaks, maybe tulip poplar, which are a more southern species as well. And we want to do that with large patch cuts where we feather the edges of them over the years. And we're going to center those patch cuts on high mortality areas. This is a map of the spots chosen to do each of these treatments in. So that little white arrow is where you come in on the hiking trail, and the white line is the hiking trail going through the sites. The resistance area was chosen because it had a chunk of conifer trees in there, so it had some more unique habitats in it that we wanted to protect. The transition area in orange was chosen because it had the highest mortality of our treatment areas. There is a matrix in between all those treatment areas that is the rest of this oak hickory stand in Mohegan State Forest. And it suffered a lot more mortality than the spots we chose for these scientific experiments. So a lot of that was sort of silviculturally clear cut to regenerate the oak and hickory, as most of the canopy had suffered a lot of mortality. I'll show you some pictures of that in a little bit. Okay, so this is our resistance treatment. We wanted to maintain oak and hickory. We wanted to start this shelter wood cut that's not going to be finished for another 20 years. And we wanted to protect our reserves and our unique habitat. So you can see there's a lot of debris on the ground here. A lot of cutting was done, but a lot was left up as well, especially a lot of those conifers. There's a lot of hemlock and some pine in there too. So we wanted to protect those spots. This is our forest crew wandering around. So we deep Connecticut Deep is the manager of this forest, and they bid out the project. So this was being done by professionals, not by our undergraduate crew. But our undergraduate crew did get a good chance to look at the whole operation and check out the work that was being done there. So you can see we tried to maintain the multi-age cohorts in this area. Also took down multi-aged trees. All right, so sorry, our stocking guide on the side here. Okay, these line graphs, that the darker line in the background is the existing conditions, and the red line is sort of the existing conditions with the mortality removed. And the blue blob is the intended treatment. So that's what we were going for with this resistant treatment. And if you can see the darker blue line in front of the blue blob, that's kind of where we wound up. So you can see with this treatment, we were kind of spot on where we wanted to be. As far as trees per acre, it looks like we might have taken out a little more than we meant to, left a little more basal area in the higher DBHs than we meant to, but pretty much spot on. The multicolored lines underneath that dark blue line represent different species. So you can see that we have a good mix of species still remaining in that stand. This is our resilience treatment. So we wanted to do these patch cuts here to encourage multi-age regeneration. So these photos were taken in December, no leaves on the trees. Can't see what is dead or not dead. But you can see we tried to create these canopy gaps, these big patch cuts in this area. That'll also open up a spot where we can plant our blight-resistant chestnuts, ideally. We're working with the American Chestnut Foundation on that. We should have seeds coming in soon, so that's going to be exciting. It's a little tough to really see the differences because I don't have photos from the same season year to year yet. But you can see we did take this density down quite a bit. But there's still a lot of trees remaining in that resilience area. A lot of coarse woody debris on the ground. Some of this might be before a lot was skidded out, but we did want to leave a significant amount of coarse woody debris on the ground, both for mud protection as the skidders are working out there, also for habitat, and also to, grind that carbon right back into the soil where it belongs. This is the skid trail through the resistance area. Our applied forest ecology lab got a chance to go out there recently and see the work as it was being done. So this harvest, I think, is nearing completion. They're done with almost all of the sites at this point. So this is the resilience prescription. The green blob is what we were shooting for there. And the dark green line is what we wound up with post-harvest. Our undergraduate crew, like I said, did not get out there with chainsaws, but they were out there doing prism plots as the professionals are finishing up this harvest. So that's where this data comes from. So you can see here, we've got a bit more left than we were shooting for, especially in the smaller DBH area. And we have quite a mix of species still left there. So we did want that species mix, absolutely. But I'm going to talk a little bit about what we're going to do with that extra smaller DBH stuff in a bit. So the transition treatment, the transition area was chosen because that's where we had the most mortality. So we wanted to put our big patch cuts where we already had big canopy gaps going on. So we had a lot of that oak mortality, but we also had a lot of advanced regeneration in all of these sites, really. So that the big open canopy gaps really gave a chance for that advanced regeneration to get going, and that should meet our objective of getting oak and hickory regenerated in this area. So big patch cuts, big opportunities for planting in this spot too. So you can see difference from the resilience photos. This almost looks like we left a few behind as opposed to we took a few out. Big open spaces in the transition zone here. This is our applied forest ecology lab, wandering around having a look at the work. And this is our forest crew on the right-hand side doing a post-harvest inventory. Most of the stuff had been skidded out of the transition zone by the time they were doing this inventory. These were our goals for the transition treatment. That orange area is what we wanted to have left. So significantly down in the understocked area there. And this is what we wound up with. Once again, we got a bit more left than we meant to have, and all in the smaller DBH area. And still a mix of species left there. But we should have a lot of smaller oaks left, and those are the ones that are going to start filling in the canopy in the future. So we have a lot more of the smaller DBH trees left than we kind of meant to. But that's because we're going to do this thing that Tom's probably going to get into a little bit during his talk. Just a little preview here. We're creating like a slash carpet to protect the oak trees from deer and give them a chance to get going. So give that advanced regeneration that's already there a chance to get going. And the crew laid a little bit of the slash carpet prior to the harvest in the transition zone there. So that's the way that we're going to go back and take out those smaller trees that got left behind during the harvest. And we're going to create these slash carpets with them. This photo is not as the orange flags that are all over it are not as visible as I was hoping they would be. But this is Tom Worthley showing off a bunch of advanced oak regeneration hidden under that slash carpet. This is the landing zone. Like I mentioned, the matrix between our experimental sites had a lot heavier mortality than the sites that we were actually working with. So what we wound up with, what Deep decided to do in those areas was pretty much take out everything. So clear the site to give the oak advanced regeneration, and there's plenty of seed trees around the area. So we're going to get those oaks and hickories back regenerating in this area where the drought gypsy moth, drought spongy moth combination had taken a lot of them out. And going forward, an important part of this whole experiment is going to be the monitoring that we're doing. So where am I on time? I got a little time. So we put 4 random plots into each of those experimental treatment areas. So most of the data that we have for stocking comes from just doing prism plots wandering around. But these 4 permanent plots are going to be the way that we're going to look at the forest response to these treatment options. So there's 4 little pink dots in each one of those experimental zones there. And each one of them is this multi-layered monitoring plot. So the larger circle, we're going to keep track of the overstory trees, and there's 3 small circles there where we keep an eye on the saplings and the smaller trees. Little squares where we keep track of the ground cover, and transects that we keep track of the coarse woody debris with. So we're going to watch all of that stuff going forward and really be able to see how the forest responds to these treatments. One thing that we talked about in the workshop was questions that people had about how the forest would respond. So built into this monitoring plan is a way to look at all of our local concerns. Whoever had a question that they were hoping that this experiment might answer, we built it into this monitoring plan to try and figure out the answer to those questions. So, and that leads me into talking about our replicate sites. So we're working with Christopher Riley at URI to put a replicate of this whole thing at Tecopit Forest in Rhode Island. And there are some local differences there. So we're going to run like a mini workshop. We want to use the same treatments that we used in Mohican State Forest, because, you know, we want to replicate the science experiment, but we do have local concerns there. So we want to run a mini workshop to make sure that we get everybody's thoughts, opinions, and their questions that come from local conditions and local things that are going on. We want to get those answered with these plots. So we got a similar forest, oak hickory forest type going on. They also had a similar mortality event between the spongy moth and the drought, a lot of mortality of oak there, but like a lot more actually than we saw at Mohegan State Forest. So the treatments will have to deal with the fact that there was a greater severity of mortality there. We also have a lot of cultural cultural elements to consider in this forest. There's foundations and other things like that that we're going to have to plan around. So that's exciting. And this has just as many invasive species problems as Mohegan State Forest did, perhaps different species. So that's one replicate site. And a second replicate site that we're doing is going to be on Yukon Forest. And we're working with the undergrad students. Andrew Muller is on this one right now. He's mapping this site and dividing it up into the different experimental treatment areas. So once again, 5 to 7 acre experimental plots, and we're going to run a resistance, resilience, and transition treatment in them. So some other things to consider around this one is it's adjacent to privately owned property, so we have to deal with that. It has a similar invasive species problem, similar forest types. In the resistance treatment, where we want to preserve those unique habitats, unique species mixes, I think we have some pitch pine pockets there instead of the hemlocks. So it's going to be a little different in that respect as well. But it's going to be a great replicate for the Mohegan project. And I think that's all I have. Yeah. So thank you all for listening to me, and please let me know if you have any questions. I think I got a little time.
Great, Amanda. Yeah, we do have a few minutes. I have a question or two.
By all means.
I noticed that you did include mention about southern adapted species, and I think that that's going to become a little bit more important when it comes to replanting some of these areas. We're going to have to look at some of the species that are better adapted to hotter, maybe drier conditions, similar to what they have in the South. And I am glad that you also mentioned consideration of invasive species. Could you comment? Are you going to be managing invasive species to a large extent, or just recording what they're there, or how are you going to deal with invasive species?
They are definitely going to be managed. I kind of left them out of the prescription descriptions for this one because it's going to be the same for all of the treatment areas. They are going to be managed. Mohegan State Forest is a Connecticut state forest, and all of their management plans. We did write this experiment into the official state management plan for Mohegan State Forest, and they wouldn't do one without dealing with the invasive species. So there was a pre-harvest treatment to deal with. It was mostly multiflora rose in Mohegan State Forest, with some barberry by the wetter areas and things like that. So they did a pre-harvest treatment, and they have money set aside to follow that up with it's a certain amount of invasive species treatments that they will be following it up with, and we're going to have to replicate that in the other sites as well. So yeah, invasive species, we are going to attack them with everything we got. Does that answer your question?
Yeah, that was great. Yeah.
Cool.
JP has a question in the text box.
Okay.
Let me see here.
Can you read it out loud? I'm not
Yep,
sure.
I'm getting there. JP's asking, might there be any concerns about assisted migration of southern oak species and hybridization with native oaks from the legal perspective within the state of Connecticut agencies? There's a potential to create novel species which may fall under protection.
Yeah, so those things came up to us, I want to say theoretically, when we were thinking about all the things that could happen here. But part of the social science aspect of this is seeing if anyone else thought of it. So in our initial workshop, that did not really come up as an issue. People seemed mildly hesitant in some cases to really to really get novel species mixes going on up here. I didn't hear anyone bring up the idea of novel species, but that's certainly a possibility. But I guess we're waiting, and we're keeping an eye on the social context to see if anyone is concerned, if something happens, and what should be done about it. So it's in the back of our minds, but hasn't really been brought up as an issue yet.
Okay, there's one other, and Bob Fahey commented, my good friend Andrew Hip, who studies oak genetics, refers to oak individuals as bags of genes, which meaning species boundaries are very fluid to begin with. I've heard oaks referred to as very promiscuous, so bags of genes is a little bit more polite. So
I've heard promiscuous as well.
yeah.
Yeah. Yeah, I mean, it's definitely something to keep in mind that for the transition treatments, especially, we want to encourage the forest to be able to adapt itself. The forest needs to sense what's going on, and, you know, using our help, using the boosts that we're giving it, it needs to be able to figure out what it should be to deal with what's going on. And if that involves creating new bags of genes, then so be it.
Hey, Amanda,
Yeah.
one question is, I may have missed something critical when you were discussing resistance, which I believe you mentioned is basically maintaining the oak hickory and moving towards a shelter wood. But why did it take 20 years to complete that shelter wood rather than more of a textbook time frame? Is that because initially it's really just more of a pre-shelter wood thinning type work?
Yeah, I might not have been super clear about that. What we've done now is a pre-shelter wood thinning cut, and we're planning 20 years from now to maybe remove the overstory. And we built that time frame to be pretty long. I think because in some ways we want to see what happens. So that area suffered some mortality of oak also. So we want to be able to make sure that the oak and hickory regenerate. But if there's more mortality to come, you know, we might have to alter our plans as we go. So that area we didn't want to mess with too much just yet, and we wanted to give it some time to really see what might happen with it before we absolutely settle on what we need to do about it. Does that make sense? Okay.
No, it makes sense. Thanks.
Yeah, one other question in the chat. Rick Shank wants to know, what were the demographics of the original workshop? It seems odd that hunting was so far down on the priority.
Yeah, so that is definitely something that I felt like we didn't do a super great job at. Ideally, you want to get all of the local stakeholders involved, and we didn't get a hunter on our workshop. And I know they're out there. We see signs of hunters hunting in that forest. Hunting is legal in Mohegan State Forest. So that's definitely a piece of the puzzle out there. And we've been keeping it in our minds. We know people do that. And we want to, maintaining the oak hickory component is certainly for, in some ways, to maintain the wildlife species mix that's out there. But yeah, we did not get a hunter on our workshop. The demographics, it was mostly forest academics and the folks from DEEP and local land managers. A lot of forestry academics. So a lot of people that were trying their best to think about what other stakeholders would be thinking, but less of the actual other stakeholders. But that is another reason we're doing the mini workshops in our future replicate sites in Rhode Island, and we'll do another one at Yukon Forest on the Lee Farm tract. We'll do those mini workshops to try and get more people involved, get more ideas, and make sure we're covering our bases. Yeah, so that survey of workshop participants was a lot of forestry academics and actual foresters as opposed to hunters.
Thanks, Amanda. If anybody else has any more questions, you can put them in the chat. Amanda, if you could stop sharing your screen, we can get Young Hao queued up here.
All
Young
right.
Hao, are you ready to roll?
Yes.
Okay, go
so
ahead
I
and
will
share
try
your
to go
screen.
there. Okay, just a second. Is it okay coming out the full screen?
That
Vicky?
looks good. Thank you.
Okay, thank you, Vicky. Good morning, everyone. So I have been working in the Plant Disease Information Office for over maybe 12 years right now. So I just want to give you some highlights in 2021 tree disease from our office. Every year we handle about over 1,000 plant inquiries in our office, normally from the visitors, mails, emails, and the phone calls. Yeah, in 2021, we got about 3,464 inquiries. Among them, about 1/3 of the inquiries tree-related inquiries. So I just have a table just to give this about 7 mostly concerned tree issues. So this is spruce, beech, arborvitae, maple, pines, apple, and the oak. But I just want to mention that most of these inquiries coming from arborists, landscapers, homeowners, municipals, and some from the foresters. So this is the number of the inquiries, maybe about 70 and over 100. But normally, inquiries related plant health issues about over 60%. So that means most of the inquiries related to the plant disease problems. Yeah, this is first that I just want to mention that's the spruce. That's the most received inquiries. So that's the most common problems on spruce. That's a needle cast. Normally there's 2 common needle casts. One is Rhizosphaera needle cast. Another one is Stegmina needle cast. So that's the most received inquiries. And there's some that's the rust and also some canker issues. On beech, so you can see that about maybe 85 inquiries beech leaf disease related. So this is the sum, this is like anthracnose last year, because we had a little bit dry, although we had a little bit wet early spring, but that's not during the spring, we got this dry condition, period of dry condition. So that's why it might be not too many anthracnose samples we received, but most of concerned is beech leaf disease. On arborvitaes, that's the most common problems. That's the Pestalozzi needle blight. That's the fungal needle blight, very common issues. Only one issue is about the Phytophthora root rot concern. On maples, so I just mentioned there's a lot of samples coming from the arborvitae. So that means that most come from the landscapers. That's a powdery mildew is the most received inquiries because there's one that's the Japanese maple, very vulnerable to this powdery mildew. So that's why there's a lot of the powdery mildew, but not too many like the leaf spot or anthracnose. Normally, I think every year we received a lot of samples anthracnose on maples, but the last year we didn't receive. But just like I mentioned on the beech trees, so because it's a short period of the dry condition last spring. On pines, here that's I combined with a different kind of the pines together. So that's why Diplodia blight, they come out more inquiries. So that's 8, that's the Diplodia blight issues on pines. And some needle cast, and the one that's a Botrytis canker. On apple trees, most of that of apples that come from landscapers. So that's obviously that's the seed apple rust is commonly received inquiries. Some leaf spot, and also scabs. So last year we received a lot of inquiries about the scab issues. So people call in and send us some samples. So on the oak, that's the most received inquiries. That's the anthracnose, and also there's people concerned about that bud rot. So a lot of the mushrooms coming out at the base of that the trunk, because that's the tree conditions. So people concerned about it. So that's the 7 trees most received inquiries in our office. And last column, you can see that I labeled as abiotic. Abiotic, that means most of the tree condition issues because it's environment issues, cultural issues. And also some of them that's undetermined. So that's some sample come in, we cannot determine what kind of pathogen, and also might be we didn't find any pathogens on there. So I included those inquiries in this area, in this column. So that's why that's some maybe that's the high numbers of the inquiries included in this area, in this column. So I just mentioned earlier, so this is the beach inquiries are normally not too many. So you can compare this to 3 years, last 3 years. In 2019, we received about 27 inquiries about the beach. Among them, there's 2 inquiries, excuse me, 2 inquiries about the beach leaf disease. Because it's 2019, we first found confirmed beach leaf disease in our state. So that's one inquiry coming from email, another inquiry coming from sample, and also I did site visit. And then you can see next year, so that means 2020, we received more samples. So about the 60 beach leaf inquiries, and then 20 inquiries concerned about that's a beach leaf disease. And then we received the samples confirmed that's in 7 samples. But the last year, you can see that the number increased dramatically. So you can see this inquiries concerned about 166 beech tree inquiries. Among them, that's the most of them that's the concern about the beech leaf disease. So I think Dr. Barbara will talk about that more. So I just gave that the numbers update. So you can see that the people concerned a lot about that the beech leaf disease. Septoria leaf spot on maples, that's it's a minor disease. Occasionally we find some samples confirmed that's a septoria leaf spot on maples. But last year, one arborist sent me this picture on the left. You can see this is 3 large Norway maples in this landscape. So you can see that's a turn brown. That's in September. So first I thought might be that's a drought condition, but I tried to recall that we didn't get the too dry conditions during the summer. We had a lot of rain last summer. So, and then I asked the samples, and then he sent me sample. I found that the small, like on the right upper corner, so you can see that the brown spots. That's totally different from the drought stress or leaf scotch. And then I can see on the right bottom, you can see that the white spores coming out. That's from the brown lesions. So that's a septoria leaf spot. So just from this case, so it's a minor disease on maple trees. But I'm thinking might be that this strain of the Norway maple, it's more susceptible to this disease. So that's I'm still cannot confirm that's why this is the 3 large trees. That's very evenly severely damaged by this septoria leaf spot. And also on the late season in October, I received this sample. So look like the anthracnose, but this is not. This is tubercular leaf spot. So you can see this is normally little large round shaped lesions. And also you can see that the black fungal fruiting bodies on this is the necrotic tissues. So that's the black fungal fruiting bodies on there. So this is in the late season. Yeah, this is a giant square. So that's from the homeowner. So that's the two pictures on the left. So you can see compared that in 2013 and the last year 2021. So that's a 2013 sample. I found it's pictures from the Connecticut notable tree list. So this is in 2013. This is look like gorgeous. This is very nice looking. But the last year, this homeowner sent me that's the second picture. So you can see it in 2021, almost the half of the canopy on the lower part turned brown. First, I thought might be the salt spray because this area this is very close to the water. So in New London area. So that's a possible that might be the salt. And then the homeowner sent me sample. I found it's the browning needles and also that's the fungal fruiting bodies on that's the brown needles. So this is a Cercospora blight. In few years, I received some samples Cercospora blight, but not damage like this. So this is the so severe damage on this tree. So because it's a fungal, this is a fungal disease already on the tree. So that means that the next few years, they can spread from the bottom to the top. So I very concerned about this. On Eastern Red Cedar, so last couple of years, we received a lot of inquiries associated with the browning of needles because it's a drought during the season and the warm winter temperature causing a lot of the stress on the Eastern Red Cedar. But last year I received one sample, look like the browning of needles on the left picture you can see. But if you look closely on the right picture, you can see on the brown or black stems, you can see that the fungal fruiting bodies coming out. Actually, that's the fungal canker or blight, Phomopsis blight on this tip. So that means that the stem damaged and then tip turned brown. So look like that's the environmental stress, but it's the fungal disease. This is the pitch pine tree. So normally we received on Mugo pine trees, Diplodia blight is very common, but this one is I first received this sample on the pitch pines. So it's a browning, sometimes maybe just the tips, and also some browning of just the needles on the right. You can see not just a whole needle, just a tip or part of the needles and the new growth turned brown. When I look at that under the microscope, I confirm this one is the Diplodia blight. So on the pitch pine.
Young Hao, you got about 2 minutes.
Yes, okay. So on the white pines, yeah, normally Carnivorgilla needle cast, this is, we saw this is a couple years ago, but this is the Bifidella needle cast. Last few years, occasionally we receive this sample. So this is, you can see on the left picture, it's the turn, some needles turn brown. And then when you look closely, you can see on the right picture, so you can see black fungal fruiting bodies in the middle of these little brown needles. On spruce trees, I mentioned that the Rhizosphaera and the Stigmina needle cast are major issues, but this is the trauma needle blight. So this also you can find on the spruce. So on the left picture, so this is different from this the Rhizosphaera needle cast. Rhizosphaera and the Stigmina needle cast normally turn, most of the needles turn brown, but this is the trauma needle blight. Only you can see on the few needles on the left picture, you can see, and also not the whole needle, just a part of the needle you can see on the right picture. Parts of the needle turn brown, and also obviously you can see there's a fungal fruiting bodies in the brown tissues. I think that's my last slide. Okay. thank you. So I don't think we have any time to take questions. Okay.
Yeah, great, Young Hal. It was a
Mhm.
great summary of the things that come across your desk. If you have any questions or comments for Young Hal, you can leave them in the chat.
Okay, I will
Um,
check that.
Serves as a good segue for Bob Mara, our beach leaf disease expert. Bob, if you're ready to go, you can start sharing.
I am ready, and I'm assuming you can see my slide.
It looks lovely. Take it away.
Wonderful. Okay, so this is kind of just an update, really from an update from last year. I just have a few more slides to show, so we'll get moving here if I can get my slides to advance. They're not advancing. Okay, there we go. All right, so for those of you who are not familiar with beach leaf disease much, just a very quick rundown on the history, the very brief and short history. This is a disease that was first identified in Ohio in 2012. It is now proven to have been caused by, that this disease is caused by a foliar nematode named Leidylenchus crenate subspecies McCannii. It's a subspecies. Sister subspecies is the subspecies crenate, which is in Japan. And I'll talk a little bit more about that in just a moment. Here in North America, it's infecting both American and European beaches, and where it's found, ornamental plantings of oriental beech. Oriental beech is not from what people might think. It's actually from kind of the area around Eastern Europe and the Levant. So it's not oriental in the sense of being Far Eastern Asia, just to clarify. But that is also a very susceptible host. We don't see it really very much around here. These are micrographs from USDA nematologist Lynn Carta, the paper that they put out in 2022, paper where they demonstrated and proved using Koch's postulates that the nematode is causal. Right now, we know that it is necessary, and whether it is sufficient, meaning if there are other agents that are also necessary, is currently under investigation. But this is what the nematode looks like. These are very small, very easy to isolate in the lab if you get them at the right time of year, which like around, we can get them out of leaves by taking leaves and floating them in water. Starting in, the earliest would be late June. Before that, it's very, very low frequency of extraction of the nematode. That fact, given that the leaves emerge fully symptomatic and there's a delay of a couple of months before you're actually going to see the nematode, is what led researchers reasonably, justifiably, into exploring other causes other than the nematode. And I'll talk again a little bit more about that in a moment. The disease progression, the leaves emerge fully symptomatic. So in this upper right-hand picture, you can see what the symptoms look like. Darkened bands, sometimes you'll see hypertrophy, but they emerge fully symptomatic, and the symptoms you see upon leaf emergence are the only symptoms you'll see all season long, which is probably not an indication of how the nematode is spreading. When the nematode moves mid-season into other portions of leaves and other leaves on other trees, it can set up housekeeping in those leaves, but not cause these symptoms. Late season, the banding often thickens, and it really depends on how it interacts with various environmental conditions like drought or heat. And so you can see more or less of the kind of symptoms that I'm showing in the lower right-hand corner. And in subsequent seasons, we're seeing aborted bud development, thinner canopies, especially in the saplings, and mortality is observed in some saplings in 2 to 5 years. So this is in that left-hand side view, you can see looking at the underside of the leaf, holding it up to light is really when the symptoms are most dramatic. But you can also see in the right-hand side towards the tip of that upward pointing leaf, you can see the hypertrophy. So the exaggerated growth of those two bands. And looking at it from above, again, this is what the hypertrophy looks like And you can almost make out that darkening as well, although it's really most dramatic when you hold the leaves up to light. All right, so end of season 2020. This is what we knew about based on survey work and based on, we actually all on survey work and reports that were then later confirmed. This is what we knew about the distribution of beech leaf disease. And when I say confirmed by proving the existence of the nematode as well. And then end of season 2021, the map now looks like this. And I just want to caution that we not assume that everything we're seeing here is the result of spread. It probably is to some degree, but it's also the result of increased scrutiny and also more better ability to identify when it's seen. So we can't attribute all of what we're seeing in these changes from year to year to just spread, but also, as I say, increased scrutiny. So that's what we know as of the end of the season 2021, and what we can expect for 2022. Probably we'll see it maybe further west in Massachusetts. I know that the folks at U.S. Forest Service in New Hampshire and Durham looked very hard in New Hampshire, didn't find it, and in those southern coastal counties of Maine and didn't find it, but we'll see what we see. And you can see it's actually confirmed as far south as that northern part of Virginia. So whether or not it's also in other parts of Maryland, we'll find out. Okay, the life cycle of the nematode, a little bit about it. Then we know the nematodes overwinter in buds. I have not succeeded in getting them out of detached leaves on the ground, although that was reported in a paper last year, and certainly not on attached leaves. And I've been looking this year, but when I go to trees that I know were infected last year, this year I've been bringing buds back to the lab, mostly because I need nematodes for the research I'm doing in my lab. And I have no problem isolating very viable nematodes, both juveniles and adults, out of buds. So they survive even those very deep cold spells perfectly well in the buds. Just prior to bud break, we don't find the nematodes any longer in the buds. That doesn't mean they're not there. It's just that there are no longer any viable nematodes in the buds, and these nematodes are not long-lived. They persist through multiple rounds of reproduction. In spring, at leaf out, fully symptomatic, but we do not get nematodes out of the leaves. And whether that's because there are no living nematodes, perhaps only eggs, because PCR signals that I use a marker that I developed, a very easy marker to deploy that is highly specific to this species of nematode, confirms that it's probably eggs in the leaves, or maybe eggs and or recalcitrant adults. We just can't determine which of those is the case. But what we do know is that starting in early July or maybe late June, we will start having no problem encouraging nematodes to exit leaves into water in the lab. And we also know, based on a paper that came out last year, that the population densities of the nematodes increase into August and continue to increase into September. So they're going through these rounds of reproduction inside these leaves, and really, really getting crowded in these leaves. So much so that in a paper by Lynn Carta and Gary Baushan and others, they produce these micrographs in the upper left-hand corner. That's what the cross-section of a healthy beech leaf looks like. You can see the mid-vein bundle here. I don't know if my pointer is showing up on my screen or not, but that middle section there, and you can see the spongy parenchyma and the mesophyll. And in the lower right-hand corner, from an infected symptomatic leaf, what you're looking at are, in addition to a lot of degraded plant tissue, you're looking at the casts in this micrograph of nematodes. So they get really crowded. And I think that when we bring these leaves into the lab and put them in water, there's a mass exodus for the exit door, which in the case of the nematode would be the stomates. So the more crowded they are in the leaf tissue, the more readily they exit. And that is how they actually get around, at least, locally on the leaf. As soon as there's any water film on the leaf, we know that they exit the leaf, and there could be a rain splash, could get picked up by an arthropod or an animal, and then transported over to being near a newly a leaf that's not yet symptomatic, and then get into a stomate, and then set up housekeeping in a previously uninfected leaf. So I just want to, just based on a lot of conversations, I also receive a lot of inquiries about beech leaf disease, and I've been giving a lot of talks about beech leaf disease. And I just want to confirm, beech leaf disease is caused by this foliar nematode, Leidylenchus crenate, subspecies McCannii. It is not the presumed cause of the disease. It is a cause, if not the cause of the disease. There is still no evidence to support a hypothesis, which seems to persist, that the real cause is anything but the nematode. For example, the leaf microbiome or the nematode's microbiome. And there is still that hypothesis persists, but there's no evidence to support it, other than to say that it might be the case. And there is also still no evidence to support the hypothesis that the nematode came from Asia. It is a good hypothesis. It hasn't really been explored, but I've been asked about this several times. So I just want to clarify that we can presume that the nematode came from Asia, but we have to support that as a hypothesis with evidence, and we have none. So just to make that clear. All right, and so we have long-term monitoring plots installed that were installed in 2020. I put in 11 of these, 1 or 2 per each of the Connecticut's 8 counties, and we measure them each year using an FIA forest inventory analysis data collecting format, and we have temperature and relative humidity data loggers installed. The original plan with the Forest Service for the entire region was that there'd be data loggers of this type installed at all the plots, but it turns out Connecticut's the only place that has data loggers installed. And these are data that we collect. We go back and remeasure the plots, and we give those data to our Forest Service colleagues. And this map of Connecticut here, the blue pluses or crosses or whatever, those are where the long-term monitoring plots are installed. We did survey work in 2020. We had funding to do that. We did not have funding to do that last year, so we did not do any survey. We only confirm positives when we get reports from people, and then can confirm ourselves that it was indeed beech leaf disease. But you can see now that we have positives in all 8 counties. Places that we looked in 2020 and did not find it are the green X's, and the red pluses are where we did find it, or where we've either found it in a 2020 survey or we've updated the map with positive reports for 2021. So there's that. And I just want to point out that beech leaf disease is still most extensive along this sort of 10 to 15 mile band paralleling the shoreline. And although we are starting to see it further north, it's scattered. We'll see when we have a chance to look. Again, we have no money this year for survey work, so there will be no survey work this year. And so other than visiting the plots, it'll be just other people who are out looking, and we'll be finding out if it's more extensively distributed and getting similar results from the other parts of the Northeast. I just also, okay, so on control, which is of course is not something that's relevant to forests, but on properties, people are concerned about how to control beech leaf disease on their properties when they have it. Emamectin benzoate and other nematicides are not showing any effectiveness. Part of the problem is getting it from the ground up into the foliage. And so that's been abandoned pretty much. But Something much simpler, cheaper, and easier to apply is polyphosphates as a soil drench. Both soil injections and drenches that were tried. Drenches seem to be just as effective as soil injections. And the folks in Ohio are seeing very, very promising results on saplings. The way these experiments were set up, they have controls, and it really does seem that multiple two applications per year. I don't have the details in front of me, but I think they were doing it in June and again in August. And just following the instructions on this one brand of polyphosphate called polyphosphate 30. And they had very, very promising results. And they're now this year and next year, they'll be collecting data on mature trees as well. And that's it. So I do have time for questions if anybody has any.
Great, Bob. That's a really nice summary. I know my inspectors have found beech leaf disease in several nurseries as well. I'm not sure what the implications of that would be, but we've found it.
On
So,
European beech.
yeah, on European beech and on a couple of times on American beech as well. There's a few nurseries that do grow American beech. So great. If you have any questions for Bob, you can put them in the chat and we'll get to them later. Next up, we have Tom Worthley. Tom, are you ready to roll?
Yes, I am.
Yeah, we got to get Bob to stop sharing here.
I'm trying to do that. And oh, there we go. Okay, there we go.
There you go.
And
So-
I'll start at the beginning here.
That's a good place.
Hopefully you can see a slide that says fresh look at preparatory treatments and shelter wood systems to
Yep.
foster oak regeneration. And I'm
Yep,
not
you need to put that in slideshow. There you go.
I-
We got it. Perfect.
okay. You got it. All right. I'm not hoping to tell anybody anything here that they don't already know. I'm hoping that we can get a little experiment started here to look at some small questions around timing and site conditions to foster oak regeneration where we already have it on the ground. And so this is a very small nuance associated with these shelter wood systems that you've heard about already this morning. And hopefully in the process, I'll be able to reinforce a couple of the points that you heard earlier from Dr. Ward and from Amanda in the course of the presentation. We have advanced oak regeneration. It exists on many upland oak sites. These are little seedlings that we find across the forest floor on many of the upland sites that have come from years when there were abundant acorn supplies and something didn't eat all the acorns. And sometimes these seedlings have been there for years and years and years. Dr. Ashen down at Yale talks about a massed year in the mid-1990s that produced a lot of oak seedlings that just have not developed into saplings or whole-sized trees yet, but still remain in that kind of seedling size. And the problem is that they get shaded and they get browsed regularly by the deer. And so those are two challenges that we have in terms of bringing along oak regeneration where we would like to have it. The shading not only comes from the upper canopy, those parent oak trees that we have dominating the canopy in very many places, but the fact that we have a layered effect in many of our forest stands of shade tolerant hardwoods that create a second layer under the main canopy. And these are stands that we refer to as even age stratified mixtures. A lot of those smaller trees, not actually younger trees. They're trees that are just as old as the main canopy trees. They just have not grown as fast because they are tolerant of shade. They exist in the subcanopy. We see oak regeneration quite often, like this photograph on the left, you know, in the woods. And if you step back and you look at where that photograph was taken, you can see that the shade can be quite dense in some places, preventing those seedlings from developing much more than they already have. Oak is one of those species that requires adequate sunlight in order to develop in height and so forth and so on. This is a photograph taken some years ago of an 80-year-old oak stand. It's got 5 different species of oak present in the stand, and we took some inventory of that, and which I'll share. Foresters are familiar with this sort of table. We call it a stand table, and it presents the inventory with the diameter, the DBH, if you will, arranged on the left-hand column, and the species across the top. And you can see numbers of trees per acre from a sampling of the stand in each species and size category. And we can see just from looking at the table that the oaks in this stand, the white oak, red oak, black oak, scarlet oak, etc. There happens to be chestnut oak out there too. We just didn't happen to pick any up in the inventory. Dominate the mid to larger diameter sizes, but there's a huge number of stems per acre in the smaller diameters, the sugar maple, black birch, red maple, beech, many of which are trees that are just as old as the oak trees. Not all, but many of them are. And, you know, have tremendous numbers in the small diameter classes. And Dr. Ward talked about how a site can produce so much wood, can produce so much volume. We saw it in the basal area graphs and the wood volume and the carbon graphs that he demonstrated. And we have choices in management about whether we want that wood to be growing on one size tree or another. And so if we graph these data, oh, by the way, very often foresters consider these smaller stems to be a different functional group. They don't really compete much with the, with the larger trees, and so don't often consider them in the inventory. And so they look at numbers of trees 4 inches and larger, and that's what that red figure represents down below, 240 stems per acre of 4-inch and larger trees. But if we graph the data, you can see it quite vividly that there's a huge number of small stems compared to the mid-size and larger stems that we have. If you look at the classic reverse J-shaped curve that we're taught about in forestry school that represents an balanced uneven age stand, it begins to look pretty similar. We can argue about, do we truly have a functionally uneven age stand here, because a lot of these stems have a long life expectancy and can persist in the shade for a long time. But we do refer to these as even age stratified mixtures. And while they look very similar to uneven age stands, it's not exactly the same. Basal area can also be displayed in a similar chart. We can see the numbers here rounded off to whole square feet, and representing 112 square feet per acre in this case of trees 4 inches and larger. The small stems don't add up to much basal area, as we can see in the graph. And while there's a huge number of stems, they don't contribute much to the total basal area. But that's not my concern at the moment. If we plot these data on the Upland Central Stocking Guide, we can see that this particular stand falls right at the the overstocked, fully stocked to overstocked condition. It's if you were to walk around out there, you would see some trees dropping out of the picture from competition. And a more desirable place might be, lower density, lower number of stems to grow the trees that I would like to grow. And if I'm interested in growing oak trees, then it's the oaks I want to work with. These are the trees that I want to have as my seed source, as my shelter for seedlings, especially if I have any advanced oak seedlings on the ground. Okay, and so the smaller diameter stems and the mixed hardwoods that are shade tolerant very often get ignored when we talk about silvicultural systems, and like they'll go away or something like that, and they won't necessarily do that. If we have a disturbance like Lamiatria dispar or spongy moth that eliminates half the oak canopy, black oaks that drop out of the picture and so forth and so on, then we're going to see those get replaced by the shade tolerant hardwoods and so forth and so on. So looking at my stand table again, here's the stems I want to work with. Looking at the basal area, these are the stems I want to work with. And here's another example that you saw earlier from Mohegan. Once again, we have that, even if you take out the dead stems, we have that kind of reverse J shape, large numbers of small stem condition from our inventory, it being an even age stratified mix. Here's a picture of this subcanopy that survived the defoliation and drought by the spongy moth, and is poised to take the place of the oaks in the upper canopy, and it won't give our little seedlings a chance. So the issue, of course, is perpetuating oak communities. That oak regeneration is challenging because the acorns don't stand a chance. The birches and maples produce millions and millions of seeds every year, and the birch, if they don't find a good opportunity to germinate, can hang around in the soil for 5 or 7 years and wait for their chance to germinate and grow. The acorns get one chance if something doesn't eat them first. The seedlings are browsed and shaded. And so when we talk about silvicultural systems, and we talk about the application of a shelter wood system, in oaks, we're talking about bringing along that advanced regeneration under the canopy of the existing oaks. So first of all, remember, it's a system. We're managing this from the beginning to the final harvest, and starting over again, and doing all the steps necessary. One treatment in a forest is not silviculture. We need to apply it as a system. In the shelter wood system, our regeneration is sheltered. A new stand is established under the partial shade of the older stand. The removal of the older stand occurs in several stages, and the new stand is tended as an even age stand. Shelter woods can come in many varieties, 2-stage, 3-stage, irregular. You'll hear all kinds of descriptions. But this initial stage, especially where you have regeneration already established, is called a preparatory cut. At the risk of sounding a little critical, but I am the professor after all. It's not a pre-sheltered wood thinning. It's a preparatory cut. We remove the undesirable growing stock, and we create some space and some sunlight. And if we have advanced regeneration, then we are in sheltered wood mode, okay? So the nuance I'm talking about is focused on places where we have advanced oak regeneration already. We want to eliminate the undesirable growing stock from the stand, as the classic shelter wood system suggests. And we want to eliminate the subcanopy shade and increase in ambient light. And I'm talking about eliminate. I'm talking about getting rid of it all. We want to protect the oak seedlings from browse, for a year or 2 or 3, perhaps, if that's possible. We'd like to add organic material to the soil if we can. And so we want to also design something that perhaps a private landowner will actually do. And so my idea here is to recommend preparatory cuts and create a slash carpet, as Amanda described earlier. Now, Emery Gluck has done a good job of showing us over the years of eliminating sub-canopy shade using controlled burning. But what I'm going to suggest here is that we go through this step of felling everything that we don't want to actually be growing and spreading it around so that it protects those seedlings that already exist from deer and eliminates sub-canopy shade. I basically want to take what I want to grow, I want to eliminate everything else and turn it into this. And hopefully we will get some protection from the deer. And for 2 or 3 years, the material will rot down sufficiently so by the time we want to do an upper canopy disturbance, it won't be a hindrance to operability. I tried something like this over in a post-harvest situation on a portion of Yukon forest, and we have very robust oak seedlings. They're certainly competing with everything that's sprouted up. They're very abundant. They're getting plenty of ambient light. This was an irregular variable canopy retention type of harvest, but we took some funds from the harvest and we spent it on going around and dropping all the ironwood, dropping all the beech, dropping all the black birch and all the red maple, and just leaving it as a big carpet on the ground. And eliminated a fair amount of deer browse for a couple of years. And so these seedlings were able to gain a little bit. And so here's my hypothesis. In places where we have advanced regeneration, this preparatory cut with a slash carpet will provide deer browse protection for 2 or 3 years, will increase ambient light sufficient to observe some seedling response. It will decay sufficiently to permit operability for a shelterwood second stage in 3 to 5 years, and will contribute organic material to the soil and will be something a private landowner can actually accomplish, perhaps even with equipped funding. And so we're going to set up an experiment. We're getting started on that this spring. Here's my plot layout. In the recording, you can study this a little bit more, but we'll look at untreated areas, treated areas with no slash carpet, treated areas with a slash carpet. We'll protect some areas from deer, so we have some comparisons to look at. It should be fun. I have a half a dozen sites picked out. We might be open to looking at some more if people are interested in trying this out. So with that, I understand I'm the last in your break. So I thank you very much, and I'm happy to take questions in the chat or whatever.
Thanks a lot, Tom. Very good, combined with Amanda's presentation to get some idea of what we're up against for oak regeneration. We've got a break on the schedule right now. If you want to chat with Tom, please go ahead. I'm going to go get a cup of coffee. So Claire, if you are ready to go, you can put your title slide up if you want, and we will resume at 10:40. Hey, Bob, are you still on? You have a question in the chat. I don't see you listed in the participants. Oh, there you are. So we'll get started in about another minute here, Claire.
Alrighty.
So I have 11:40 on my cell phone, which is how I've been keeping time. Our next speaker has been working on emerald ash borer for seemingly forever, but she's got some good information to share with us about the hope for biological control. Claire, you want to take it away?
Sure thing. Hopefully people are back. Welcome to this year will be the 10th anniversary of when EAB was detected in Connecticut and the 20th anniversary of when it was detected in the United States. So this is getting to be an old friend, not one we particularly want to have around. So today I wanted to share with you some of the recent news about or work that we've been doing on the biological control of emerald ash borer in Connecticut. And of course, this is our beast, which we all know, and I think 95% of you have probably seen this picture in one of my shows, but I really like it. They're beautiful. This is the map of when EAB was detected by town, and of course this is a little bit staggered just because when we detect in part depends on when people tell us about it or when we're able to get to a place to confirm it. But the general pattern is still fairly clear. We started in northern New Haven County in 2012, and it has been spreading out from that point. The ash borer typically has this life cycle, as I'm sure you know, or invasion cycle. When it comes in, it's very low levels, and then it increases exponentially. And after about 4 years after it's killed a large number of the big trees, the population will drop. And so this graph is from some data that we did this summer, collection we did this summer, all these forestry folks. It's very minor sort of a survey, but we looked at some measures of ash in 3 different stages of infestation, and these are actually at areas where we did releases. So the old are places where we first detected emerald ash borer back in 2012, and the new is 3 sites, including one at the Yukon forest that we did releases and detections out in about 2018. So as you can see, the average basal area of the ash in the older sites is quite small, and I don't know whether or not I calculated that correctly, but relatively you can see that there is a trend we would expect to see. There's more ash in the older sites than there is in the newer sites. By canopy class. Now, canopy class, I should have put up a little picture of it, but a canopy class is just basically 1 is a very healthy, happy tree, 5 is a dead tree, and the others are increments in between. And if you look at it by basal area of ash by canopy class, most of the basal area in the old areas where EAB first was infested are either almost dead or dead. So you can see that in the large orange bars on the graph to the bottom left there. The middle age sites are a little bit better, but still the basal area of the ash is dominated by standing dead trees, whereas the even though there's more standing dead trees left, or more basal area of standing dead trees left in the newer areas, there's also more really healthy trees as well. Another way to look at that is by canopy class. And for the oldest sites, the healthy trees are all very small. So these are trees that have either sprouted or resprouted or stump sprouted since emerald ash borer declined in frequency, declined in population. And whereas for the new sites, you can see that there's a more even distribution of DBH across the different canopy classes. So this, of course, is not a great situation. And as we all know, emerald ash borer can be effectively treated, but chemically, but that is not really a viable long-term solution. So of course, we have the biocontrol. And I'm not going to go all the way through this slide, but essentially it's using one organism to manage another. And targets in biocontrol programs have been all sorts of things: plants, animals, fungus, or bacteria. In this case, we're targeting an animal with another animal. And we are practicing the first major type of biological control. And this is really what people tend to think of when they think of biological control. It's the classic, sort of like Coke classic. In this case, you have an invasive pest, and the biocontrol agent is an insect or is a control agent that comes from the target's native range. And the idea is to add back some of that top down, the third trophic level mortality to the ecosystem for the pest. And this is a permanent solution. This is something that's envisioned as you release it, it gets established, and it works forevermore. This is also why it's so tricky and why you have to be so careful about it, because once you let something go in a new environment, you don't really have a lot of control over what it's doing. So parasitoids are usually, for insect biocontrol, one of the things that people turn to first. And one of the reasons, or there's a couple of reasons that are linked to the tight host range, the small host range of a lot of parasitic wasps. They tend to be linked very tightly physiologically. They can't live in other hosts. Also tightly linked with the host habitat. And up there, I could also have things like the host's phenology, etc. So they have to be really well synchronized with their host, and they have to be able to live in the host. And that gives them a limited ability to switch hosts. And that's good for a couple of reasons. One is that we don't want it to eat our native species, whatever it is we've introduced. You don't want it to be attacking our native species. And the other is that unlike a generalist predator who eats what is available in the highest numbers, they're just looking for whatever prey there is. And if there is a rare prey, they're not going to seek it out. They're going to go get something that's more common. Parasitoids are by their physiology are constrained to continue to hunt for those host target species at low population levels. So here we have our wished for outcome, that the AB has been cycling along at a very high level, an unacceptably high level, but then we know that it crashes. So while it's cycling along at that high level, we introduce those parasitoids, and then because of that, and we know that the parasitoids cannot establish and kill off enough hosts to stop that first destructive wave. But the hope is that after the EAB population goes down and the parasitoids have to perforce follow them, so to speak, they continue to hunt them even at low levels, which is what we hope, then they will hopefully continue to provide cover for new ash to come up, because they're keeping that EAB population low, even as the proportion of ash in the forest, the available food, rises. So that's what we hope. Now, there are a number of parasitoids that have been identified. There's a lot of folks in APHIS and ARS and many universities that have worked very hard on this. Currently, there is a huge emerald ash borer parasitoid production facility that was purpose-built in Brighton, Michigan, where they ship them out. But here's a picture of the little wasps that we've been releasing since 2013. So the first one I'm going to introduce you to, Tetrasticus planopenensi. We usually call them TETs. And they're an endoparasitoid of emerald ash borer from China. And they actually do provide a pretty high level of control, which is good. They will kill up to 50% of EAB larvae in China. And they have 4 generations a year, which is great. And here you can see there's the little female wasp on the upper left, and then down below, you have an EAB larvae that doesn't look like itself because it's all full of little wasp larvae. So it looks like a bag of Cheetos. And then there's just a picture of the male with his fuzzy antennae so he can find the female. Our other larval parasitoid that we have introduced is a species called Spatheus galeni. And they have a longer ovipositor than Tetrasticus, which is actually really important because they can parasitize EAB larvae in larger trees. So tets right now, they can only go, well, tets can only go into 2 to 3 millimeters worth of bark. And so they're very important in controlling emerald ash borer populations or attacking emerald ash borer populations in those smaller trees. But if you want to get them into the larger trees, then you need the larger parasitoid as well. And in my hand, there's a photo of my hand over there holding an emerald ash borer larva. And I don't know if you can see, EAB larval stuff always ends up being beige on beige on beige. It's hard to get contrast, but right here, these 2 little larvae, and then there's a little one right here. These are all their external feeders on the emerald ash borer. And this is a tunnel for EAB that's full up with the cocoons. So we've been doing these releases, as I said, since 2013. And the drill is that we do them for 2 years, and then we move on to new sites. And as the range of EAB has spread, we're always trying to sort of go on the forefront of that range. Our release sites spread out as well. And we've had, I think, 15 sites so far that we've done releases for, and this is where they've been. So the first 2 were in Prospect and Middlebury, out to Sleeping Giant and Sherman, and out and out and out. And our current site that we're just finishing up this year is up in Union. After that, I think we're done. The state of Connecticut is small. They've been establishing well. This is our, I think this end stage of the project, the releases, is coming to an end. And this is not quite a complete list because I don't have the last couple of years on here, but you get the idea. We've released a lot of wasps over the years, a lot of wasps. So one of the first questions with the biocontrol program is whether or not your parasitoids have actually established. Are they there? Do they live over the winter? Are they able to survive the winter? Are they able to, emerge at a time when they can find hosts? All those good questions. So there's a bunch of different ways we look for parasitoids. One of the major ones is very direct. We just go and peel trees. And since EAB lives right under the bark, that's a very good way to look at them, as long as you don't accidentally obliterate them. And you can see the parasitoids in the tunnels. So there's a couple of examples there with the pink arrow. To the right of the screen, there are 2 other techniques that we use. One is called a yellow pan trap, which is made of a yellow solo cup bowl. And that just catches parasitoids that stumble in. It's not a terribly efficient method for catching parasitoids, but it is very low input and low knowledge input. It's pretty easy to set up and collect. The other technique there is what we call a sentinel log, and that log has been seeded with emerald ash borer larvae of the proper age for infestation, and we put those out in the woods, and that little cup underneath the log there is filled up with floral foam and water, and that helps to keep the log alive because, of course, emerald ash borer is only interested in living trees, and the parasitoids are only interested in living larvae. So we want to make sure the system stays relatively happy for the 2 weeks that they're out, and then we bring them back and rear them in the lab and see what comes out. So here's our chart of recoveries thus far in Connecticut. The Union one we're still releasing, so there's no point in looking for recovery yet. But so far, I want to point out, you see that big blob of yellow in the middle? That's where we did a bunch of spread studies, and I think I talked about them a year or so ago. But we're finding them 20, 30 miles away from where we released them. So they're spreading very well, and that's really exciting. So they're establishing in the places where we let them go, where we did the releases, but we're also finding them pretty far away from where we did the releases. Oh, actually, Bethany should be pink too, because we found tets there in 2013, but we're finding them all over the place. Now, the last couple of years, we've been doing some studies to try to gauge whether or not these parasitoids, they're spreading, great. So what are they doing for us? So trying to understand if they're having any impact on the EAB population, because of course, that's the major goal, is to keep ash as a component of our forests, not just to establish parasitoids. So there's some life table studies that are going on in various states with my colleagues up at UMass and ARS down in Newark, Delaware. When we're looking at regrowth of ash in areas without parasitoids. And then also I'm measuring the persistence of parasitoids in areas with low EAB density. And that's more looking at, if the parasitoids are going to do that dive and follow them down and stay in an area where there's very few EAB larvae available. So actually this is some data from this past summer, from that last question, the persistent. So if you look on the left, there's a graph for EAB ash borer density, and that is sort of follows the basal area, but we're finding very low levels of ash borer, less than 1 larva per meter squared of phloem, of living phloem, in the places where EAB was first attacking, and then higher in the new areas, although none of them were crazy high levels, not the 212 larvae per square meter kind of levels that you see sometimes in the literature. However, and this is on more data needs to be collected, so I didn't even bother putting any error bars because it's all kind of error. But as of yet, the parasitism rate at these 3 sites is the same regardless of how much EAB are around. So if EAB are there, they're as likely to be parasitized in the older areas as they are in the newer areas. So that's really exciting, because if your parasitization rate is at the same level with a lower population, that means you're still continuing to control that population. And this is some data from work that I've done with Jean Douan down at ARS in Newark, and these are life table studies. So over on your left is data from some plots in Michigan that he's been working on out there for a long time, looking at population growth with tets in the environment. So that's what the measured is the blue. And then with life table studies, what you do is you look at the larvae or the insect at every stage of its life, and you say, okay, this many died from this cause, this many lived, this many, and you divide up the causes of mortality as best you can. And then what you can do is you can subtract one of those causes of mortality to look at what the population growth would have been without that mortality. And there's lots of assumptions and stuff that go into it, but I'm not going to get into that. I'd be happy to answer more questions. But so the blue is the observed mortality, or I'm sorry, excuse me, it's the observed population increase rate, growth rate that's there with the mortality caused by the Planopenis, so what they've observed. And then if you do that life table trick and take the mortality from the Planopenis out, the green is the rate of increase that the emerald ash borer would have had without the T. planense, without the tets. So as you can see, the rate of growth of the EAB is almost double without the tets there. If we look over here, this is data from Connecticut with the Spathius glini. And again, the blue is with all observed emerald ash borer mortality, what we saw in the field. over the years. And then the green is if you're removing the Spathius as a factor, mortality factor, and what the potential growth rate could be. And as you can see, the introduction of the Spathius took place in 15 and 16, and very rapidly it started to have quite a large impact on the growth rate, population growth rate of the emerald ash borer. So using this trick, you can take away some of the uncertainty around things like saying, well, just the EAB population has gone down because there's no trees left, you know, but that way you can get around that. Anyway, lots and lots and lots of people have helped with this research, and need to thank all of them, but I'd be happy to answer questions if I have any time.
Thanks, Claire. You did have a couple comments in the chat complimenting you on the maps, which I think we all appreciate. I myself, I'm a huge fan of maps. I love maps.
Me too.
So anyway, so yeah, if anybody has any other questions for Claire, feel free to put them in the chat. Otherwise, Claire, if you could stop your screen share, I will pull my talk up.
Excellent.
Hopefully with minimal fiddling. There we go. Let me get my slideshow started here. Come on. There we go, from the beginning.
Spongy moth.
You know, just as a quick aside, of everyone that I have talked to, no one likes the new name. We'll just put it that way. So, and I will refer to it as LDD because I don't like the new name as well. So I'm going to tell you about the LDD outbreak in Litchfield County and an LDD outbreak in the eastern part of the state, where we've almost always had LDD in Tolland and Windham, New London, Middlesex County. That would not be a big surprise, but it happened in Litchfield County. Now let's roll back the time machine to 2020, the bad old days of 2020. We were in the midst of pandemic conditions, so there was no aerial survey in 2020. At the beginning of the season, the Forest Service sends out what they call a FRAT, which is a flight risk assessment tool. It evaluates everything from surveyor preparedness to aircraft conditions, to fueling conditions, to pilot readiness and availability. And it puts all these into a big matrix, but included in the 2020 FRAT was COVID conditions, and that column was completely red. There was words such as extremely perilous, very dangerous, high risk of contagion, you're gonna die, you've got to be kidding, are you stupid or what? So we did not do aerial survey that year. So damage estimates were made by car. And I will tell you right now that we did not do mapping as we were driving. It was drive along, see something, mark it on our tablets, and then continue to the next spot. This was not perfect. None of us liked doing this. Most of the damage that we were able to map was from Tropical Storm Isaias and tornadoes that occurred about 2 weeks after Isaias along the shoreline. The winter LDD egg mass survey still did occur. I'll just refresh for those of you that don't know, every winter, usually starting in December, continuing till just about now, my inspectors and I go out on a 7-mile grid pattern roughly, and we basically get out of the car and look and see how many egg masses we can find. So that still happened. So our 2020 aerial damage survey data summary, we did record about 156,000 acres of mortality, primarily in the eastern half of the state. We did find a few acres, about 108 acres that were newly defoliated. We did see high egg mass counts in Litchfield County. We were very surprised by this and somewhat alarmed, but that's what we found. Couple of new things we found. There was a lot of agromyzid fly, also known as oak shot whole leaf miner, about 208 acres of damage or defoliation. I will say it took me forever to learn how to pronounce agromyzid and even longer to get oak shot whole leaf miner correct. Your tongue gets tied up in those. Hemlock woolly adelgid and elongate hemlock scale are still around, and they're causing sporadic damage. One thing that was very interesting, we found salt damage about 50 miles inland from Tropical Storm Isaias. That was just really shocking that the wind carried salty water that far inland enough to do noticeable damage to the canopy of trees. Now, here's our famous map, and I apologize because it still says gypsy moth, but that's how it is. These lines here are, again, are mainly tropical storm damage and tornado damage, especially in this area here. This little red dot here, that was still some salt damage that far inland from the tropical storm. Just to remind you, the size of the green dot indicates the number roughly of gypsy moth egg masses, LDD egg masses that we find. So you can see here in Litchfield County, we had some pretty big green dots. This was alarming. There was a large green dot here, but through most of the area where we typically see a lot of LDD damage, the dots were, small, which means that we weren't finding that many egg masses there. Now, we do this work in cooperation with the U.S. Forest Service, and they have a product called Forewarn, which is basically short for forest warning. It's a satellite interference imaging system that basically every week when the satellite passes over, it measures the change in reflectance from different areas. And we got reports about the second week of June of 2021 of something going on in Litchfield County. And we immediately suspected LDD based on our egg mass counts. Remember, that's where we saw the large number of egg masses in our wintertime egg mass counts. This is the product that we get from Forewarn. This image was sent to me on June 23rd. You can pretty much see the boundary of the state here. Up here, Litchfield County, look at that. And again, this is the change in reflectance from when the satellite passes over at roughly weekly intervals. There's also some severe changes in reflectance here in southern Massachusetts and in eastern New York, but a tremendous amount right here in the Litchfield County. Now, I did some imaging work on this to try to clean it up a little bit better, to get a little bit better idea as to where we would need to be looking. Here's Cornwall, here's Sharon, here's Torrington, here's Goshen. So you can see all these reddish, brownish areas is what Forewarn is telling us that something is going on. Again, this was about June 23rd, June 24th that I got these reports. Well, fortunately, just a few days later, we were able to get up in the plane, and this is what we saw. Well, there were large areas that were severely defoliated. We did some ground checking, and this was LDD. Just basically, you could see bare ground in these areas. This was extraordinarily surprising. We hadn't seen this much defoliation in Litchfield County in the 15 years that I've been doing this. Mostly it's in the eastern part of the state, but this is what we were seeing in Litchfield County. You can see the Berkshires here off in the distance, but entire hills, entire areas that were completely defoliated from LDD munching. Just some really spectacular photographs that we were able to get here. A lot of it was clustered on hilltops in some areas, as you can see here, but still there was a lot of acres that were severely defoliated from LDD. On our map, this is what it looks like. These are the areas here that we mapped significant defoliation. We did map some in the eastern part of the state, which is not a surprise. It's always there, it seems like. But by far, the biggest amount of damage was in Litchfield County. Now, when we calculated the areas here from the aerial damage survey, that's what the ADS stands for. The DCA is damage causing agent. And this is what our data look like. Now, I will let you know that this year we petitioned the Forest Service to add, excuse me, to add beech leaf disease to the list of DCAs, and they did that for us. We were very happy with that. We did map almost 800 acres that were affected by beech leaf disease, and we did ground truth that, and that was mostly in southern Fairfield County, where Bob stated that beech leaf disease was extremely severe. Beech leaf disease makes the trees look, and I can't describe it any other way, but they look smoky from the air. It's kind of an odd grayish discoloration from the air. The only way I can describe it is smoky. And I know that's not very exact, but that's what they look like. We've mapped over 45,000 acres of LDD defoliation, again, mostly in Litchfield County. Later in the season, there was also an outbreak of saddled prominent, also in Litchfield County. And we thank JP and Jeff for alerting us to that. We were able to map about 100 acres of damage from saddled prominent. And that remains a question. Why did that cause so much defoliation? We did get a question too about why we consider logging to be a damage causing agent. Well, like I said, the Forest Service gives us these lists, and logging is on the list as a damage causing agent. I think a better term would be a change agent, but that's not up to me to determine what the Forest Service calls it. So again, some really interesting information from that. 45,000 acres of LDD in Litchfield County, pretty crazy. Our preliminary egg mass survey for 2022, we're just about done with most of our sites. We are finding huge numbers of egg masses in Litchfield County where the outbreak occurred in 2021. Now, as you all know, outbreaks are somewhat weather dependent on the fungus that infects the larva. It's if the rain comes at just the right time and the larvae get infected with the fungus, there won't be quite such a large outbreak. So hope for rain in the spring at the right time. That's all we can say about that right now, but we are finding huge numbers, just massive numbers of egg masses in Litchfield County. So those green circles on the map are going to be very large this year. The saddled prominent remains a question mark. Is that going to cause damage again, or what's going to happen with that? And I don't have a good answer for that. You know, like I said, it's a question mark. Why was there such an outbreak of saddled prominent, and is it going to occur again? Are they going to be affected by the fungus or a virus or whatever to keep them in check? I just don't know. We do plan on doing aerial survey as usual this year, June to August of 2022. And so we're going to be pretty much back to business as usual in this year. Like everyone else, we don't do this by ourselves. Tia Blevins and Jeff Fengler, two of my inspectors, do a lot of the aerial survey. I do some as well. A lot of the graphics were made by Sarah Carson, who was my seasonal worker, who went to the dark side. In other words, she took a job with USDA. I'm deeply indebted to the Forest Service for funding this and for loads of technical assistance as well as training. They're the ones that teach us how to recognize the various damage from the air. They're just extremely helpful. We fly with the pilots of the Connecticut wing of the Air National Guard, also known as the Civil Air Patrol. These guys are great. their first priority is safety and getting the aircraft back in one piece. And I really appreciate that. These guys are really great. So that's all I have about LDD. If you have any questions, I see there's some comments in the chat. Let me see. Did the LDD defoliation in Litchfield result in loss of any white pine? There was some feeding on the white pine. They were basically eating everything. Is logging separated from land clearing? Not really. Logging as part of a silvicultural prescription is not damage. There's like I said, we get the list of the DCAs from the Forest Service and they include logging. So I guess you'd have to take that up with them. That's all I can say about including logging. I personally think that it would be better to describe it as forest change rather than logging, because that's really what it is. It's a change in the forest, and if it's logging due to development for putting in a road or for putting in housing or for putting in an industrial park, that's a forest change. So I think that would be a better description. So anyway, I appreciate your time. I appreciate the good questions, and I will stop screen sharing. And if you have any other questions or comments, we can put them in the chat, or you can just ask me. Carol, Chia, you're up next. If you're ready to go, you can start screen sharing. Carol? Oh, there you go. Take it away, Carol. I suspect you're on mute. Carol, we're not hearing you. Carol, we've got your picture just fine, but we can't hear anything you're saying. Okay, Carol, do you want to skip to Susanna if she's ready to go and come back? You can let me know in the chat.
So should I go next?
Yeah, we've lost Carol somewhere. So Susanna, why don't you go next and we'll come back to Carol.
All right, sounds
Okay.
good.
All right.
Oh, wait. Okay, can you hear me okay? And can you see me okay? Or can
Yep.
you see my slides?
Yep, we got your slides just fine.
Excellent. Yeah, so I'm gonna talk about carbohydrates and tree stress in urban trees. And one of the on the first slide, you can see a dogwood tree. So this with this weird orange thing here in the one of the branches. So what happened here is that the branch was cut off on the previous year or during the dormant season. And now during the springtime, the tree is actually leaking out some sap. And the sap is really rich in sugars, which are a very delicious food source for many microbes. And for instance, in this case, the sap is probably being consumed by yeasts that are able to store carotene in their cells, which causes the orange color on the trees that seem to be bleeding to death. The tree is not probably going to bleed to death, but definitely the loss of carbohydrates is not an optimal situation for the trees. Well, these bleeders, they're not, it's not the serious situation for the trees, but they are still losing some energy in this process. So I'm going to talk about the importance of carbohydrates for urban trees and how they can be used as a stress monitoring tool. So the role of urban forestry is pretty huge because it creates a lot of value for our society. For instance, the urban forestry and tree care sector in Midwest and Northeastern states alone is estimated to employ about 375,000 people to create 17.6 billion in industry output, which corresponds to an approximate 16 billion in payroll. So it's not an insignificant amount of money. And in Connecticut alone, the value of urban forest is also huge, and the urban foresters are effectively managing the air purification capacity worth 120 million and the carbon stock worth of $500 million, which is stored in the urban forests. So urban forests, they have a lot of, they hold a lot of value, and they have a huge importance for the livability of urban areas. One issue that urban trees face are the constant stress. Urban conditions are not definitely optimal for urban trees to grow. And with the intensifying impacts of climate change, trees are suffering even from more higher levels of stress in the urban conditions. And one of the surveys that has been conducted by the Connecticut Ag Experiment Station by Jeff Ward and Justin Alamo, who was a plant health fellows working here in 2020, this survey indicates that maple mortality in New Haven has increased from 30% to 45%. And what is concerning is that even 75% of the young red maples have died, which is an increase of 46% from the last survey year in 2010. Causal factors for this are unclear, but one of the most likely causes of mortality is the drought. So Connecticut has suffered from several droughts within the past decade, and this is likely linked to the increasing tree mortality in urban conditions. What we have also noticed is that there is disparity in tree cover between neighborhoods and how well the dying trees are replaced in neighborhoods with a lower socioeconomic status. So regarding tree health diagnostics, diagnosis methods that can detect tree health deterioration and predict tree survival would enhance tree health management. And detection of stress or trees that are about to die would actually allow us more timely application of different treatments. Usually when visual signs of stress appear, the tree is already in a very bad shape and possibly beyond repair. It's the same with certain human medical exams or diagnostic tests. Like when things start to show up in blood exams, the person is already in a pretty bad shape. One of these examples is maple decline syndrome. One of these signs is early fall coloration. And usually when the trees already start to display these symptoms, the tree is so far gone that it's pretty much impossible to do anything to put the tree back on the right track. Optimally, a good diagnostic test for detecting urban tree stress would be something that could also be applied for dormant trees, during the dormant season. which would allow more flexibility for when the testing could be done. And sleeping trees won't lie, because usually during the dormant season, the metabolic status of the trees is more stable, and we can get more reliable readings of what is actually happening in the tree. So now to the carbohydrates and stress, carbohydrates and how they relate to tree stress. So definitely it's significant that the word stressed spelled backwards is actually desserts. So you can already tell based on this that sugar has a huge significance for stressful situations. You know, we also eat a lot of sweet things when we are stressed. So there is definitely a connection between sugars and stress also in trees. Trees store their photosynthetic products as non-structural carbohydrates in stems, branches, and coarse roots. These molecules are starch and sugars, and these could also be thought as tree snacks. So how much snacks you have, the better you do. These snacks, or the carbohydrates, are very critical for tree stress recovery. This could be compared to a situation where you're heading out for a 20-mile hike and you don't bring any snacks. You won't die, but you're not going to enjoy the hike. It's going to be really difficult to kind of bounce back next day if you don't eat anything during that 20-mile hike. You're going to have sore muscles and everything is going to be a little more difficult. So if trees don't have these snacks or these storages of carbohydrates in their organs, they're also going to be suffering from stress, and it takes them a while to bounce back from the stressful situations. A significant portion of the non-structural carbohydrates are stored in coarse roots, and depletion of the root non-structural carbohydrate reserves reduces growth, uptake of nutrients and water, and predisposes the root system to freezing. This is especially seen in newly planted trees that suffer from transplant shock, because the transplantation itself is a very stressful event for these small young trees. During the transplantation, the trees effectively lose a lot of these carbohydrate reserves in their root systems. So one good thing about these non-structural carbohydrates is that they can be monitored around the year. And what has been observed is that they're most stable in the dormant season. So this kind of indicates that non-structural carbohydrates might have potential as a diagnostic tool. It kind of tells us how many snacks the tree has left, how long is the tree going to survive. It would be nice to establish these levels, like, oh, the tree has like 1 granola bar worth of energy left. And, you can kind of think about how far is that tree going to get with that 1 granola bar. So what do we know about the carbohydrate levels in the trees? Non-structural carbohydrate thresholds are associated with tree survival, and there have been a couple of studies that have actually found usable threshold values for non-structural carbohydrates to predict if the tree is going to survive or not. For instance, in oaks, root non-structural carbohydrate level of 1.5% in dormant trees has been noticed to be a relatively reliable predictor of tree death or tree mortality following spongy moth defoliation. There it is. But in balsam fir and black spruce, on the other hand, the spring foliage starch concentrations are a good predictor for if the tree is going to survive after a spruce budworm attack. And in sugar maples that are growing in stands that are not the optimal habitat for this tree species, non-structural carbohydrate levels are lower before bud break, so right in the spring, compared to trees that are growing in an optimal stand. And replenishment of root non-structural carbohydrate levels to normal can take 2 favorable years. So this situation after the stress event is very critical for how the tree is going to survive in the future. Tree species and stress severity affect the recovery rates of the snack reserves for the trees. For instance, Norway maple seems to be recovering remarkably well, which is really unfortunate because it's an invasive tree species, but it also explains why Norway maple has become such an invasive, such an issue as an invasive species. Coming up this year, an exciting new project how sweet is the life of an urban maple in Connecticut? We are going to start a new Connecticut Ag Experiment study to quantify the non-structural carbohydrate levels in urban maples. We are going to monitor certain environmental variables, such as soil and root growth space and urban heat island impacts. And then we're going to measure non-structural carbohydrates as a stress indicator. And our target or our goal or intention is to try and identify threshold values that can be used to predict tree survival and tree outcomes in urban conditions. This is a collaboration of 3 Connecticut Ag Experiment scientists, and we are from 2 departments. So including myself, Dr. Lee Whittinghill and Dr. Nubia Suversa, we are going to start this new project this year. And we also have 1 funded postdoctoral scientist position that was funded from a Louis Magnarelli scholarship. This is a like a fund that was established to honor one of our former directors. We are looking for applications, or we're accepting applications for this postdoctoral scientist position until end of March. So if you know anyone who is interested in making an impact on the health of urban forests in Connecticut, please spread the word. So a couple of questions that we want to address during this project. Do urban maples with larger root growth space have higher non-structural carbohydrate levels? And do they also have lower stress levels? Is root growth space associated with root non-structural carbohydrate levels? And are non-structural carbohydrate levels of urban maples during dormant season a practical maple decline syndrome indicator? So these are the questions that we're trying to ask or trying to answer during this project. And we're trying to address these questions by studying the association of site and tree growth metrics with carbohydrate levels in 240 urban maples, including Norway red and sugar maple, at least in 2 sites in Connecticut. So far, we're planning to conduct these surveys or studies in New Haven and Hartford. And our attention, or we're trying to identify carbohydrate threshold values for early detection of urban maple decline syndrome. And this information should be valuable for tree care professionals who are trying to address various tree health issues. Some of the urban site characteristics that we are interested in estimating is especially the available root growth space. So for instance, here you see on this slide, the image on the left, image A, there is a Norway maple that has very limited root growth space. In the middle panel, picture B, you see a Norway maple that has a lot more root growth space available. You could imagine that these trees will have very different root conditions. And because of the accessibility to nutrients and water, you might expect that these trees also have different levels of non-structural carbohydrates, or they have different amounts of snacks in their back pocket, so to say. What we are also interested in seeing if soil properties and the percentage of impermeable surface area or the urban heat island effects impact the capacity of the trees to store carbohydrates in the root systems. We're also going to look at various tree growth metrics, diameter growth, height growth, any existing mechanical damage, and physiological indicators, including water leaf potential, leaf fluorescence, and of course, the non-structural carbohydrate levels. So by looking at all these environmental, tree physiological, and the carbohydrate levels together, we are expecting to get a good picture of what is going on with urban maples and how we can better take care of them in the future. Another thing that I'm excited about this project is the chance to also try and develop a portable assay that could be used by tree care professionals for estimating the tree conditions in the field. So what we're also planning to do is to collect sap samples from the trees and measure the sap sugar concentrations to see if the sap concentrations are associated with the existing tree condition. So this could be as easy as 1, 2, 3. If it works, this is what we need to figure out if it actually works. It's also fast and cheap if it works. So how it works, or how I kind of visualize or vision that it might work. So you collect a sap sample. This can be easily done either by collecting a branch sample, and then if you happen to have a pressure bomb, you can use the pressure bomb to push out the sap from the branch sample. Another option is to just make a hole in the tree and then collect some sap that way. Next, you use a refractometer to read the sample sugar values and take the reading. And then based on the readings, we can determine when we sample a large enough number of trees, we can determine which sugar concentration value indicates stress in urban trees in certain conditions. So yeah, by using these refractometers to measure the tree sap sugars, we might be able to identify threshold values for tree stress. And then by knowing these threshold values, we can evaluate the impact of management on the sugar values as well. So this might give us like an real-time tool to monitor the impact of the management decisions that we have made on tree health. And by having these indicators, we can also predict tree survival and maybe prevent tree damage from happening. So yeah, as I said, we are looking to hire a postdoctoral scientist to work on this project. This project is not gonna, complete itself. We need somebody to take the lead on this research project, or taking the charge on studying is phenomenal. Applications are due March 31st this year. Any questions and applications will be sent to my email. And I'm happy to answer any questions that you might have.
That was great, Susanna. It's just too bad, like you said, it's too bad that Norway maples seems to be one of the better test subjects, but maybe we can extrapolate the information to other maple species.
Yeah, that's true. I'm kind of expecting that we're going to see some differences between the 3 maple species that we're studying. And it would be also definitely interesting to expand this to other tree species.
Any other questions or comments for Susanna? If not, we'll try to touch base again with Carol. Oh, there was a question from Jerry. Will you compare sugar concentration in urban trees to forest trees, red and sugar maple? So
that's a good idea. We're kind of thinking about that. We should have like a low stress urbanized site as sort of like a control, because that might give us an idea like what should be the healthy baseline, and then how the values in forest stands differ from urban trees. Yeah, that is a good point.
And another question, you probably said this, but how big of a sample size are you going for?
For now, we're planning to sample 240 trees altogether. It kind of depends like how this plays out, like how laborious it is to analyze all the samples, because we are planning to do repeated sampling. So we have several sampling time points during the season. So it's like early spring, after bud burst, and then height of or peak growth, and then later in the fall, and then of course the dormant season sampling. So it's that kind of multiplies really quickly. So even if you have like 5 sampling points, that's more than 1,000 samples that we need to process.
Yeah, and JP also mentioned you can easily find forested, non-stressed Norway maples, because Norway maples are pretty much everywhere.
Oh yeah, and they are doing so well. They are doing so well.
Yeah, and Bob Mara pointed out many trees, including maples, extend their roots beyond the planting space and into abutting properties. They do that. So,
that's true.
Yeah.
That's true. That's kind of like what we are thinking, that we can factor in the permeable surface or impermeable surface. So if there's lots of concrete or asphalt or something like that, then maybe the root systems are not as extensive. Whereas if you have a lot of lawn surrounding the tree planting pit, then the root system might expand kind of further.
Yeah. Okay, any other questions? You can put them in the chat and we can address them there. Carol, are you ready to share your screen?
Please say you can hear me now.
Oh, I got you, Carol, loud and clear.
My
Fantastic.
apologies, everybody. Apparently, it turns out my computer is so old, it doesn't have a microphone. So Lee saved me with his USB headphones. So you can see my slides, I hope. Can you see my slides?
Got your slides and we can hear you. You
Okay.
are ready to rock and roll.
I'm gonna try and rock and roll. Thank you, everybody, once again for your patience. And today I'm going to tell you what I hope is an exciting story about teamwork. Teamwork to address hemlock woolly adelgid resurgence and reinvasions in Connecticut. Now, I know all of you, okay, my slide will not advance now. Oh dear, let's see.
Hit page down.
Page down. Nothing happening? Oh my
Nope,
goodness.
we still have your title slide.
Oh, what do you do with an old? Okay, let me try again. Resume slideshow. Can you see my screen?
Yeah, we're getting your title slide, collaboration in HWA biocontrol.
Okay. Can you see the second slide now? I'm highlighting it.
Nope.
Oh,
still on your title slide.
Okay, I really don't know why it's not working. Let me try something. All right, let's see. Can you see it now?
And your title slide.
Oh, I'm on my second slide on my computer, but you can't see it.
No. Maybe you could, can you summarize without using the slides since we've got you on audio?
Oh, this is terrible. I don't understand why you can't see it. Okay, hold on. I'm screen sharing. Okay.
Yeah, we've got your title slide.
I don't understand why it's not showing. Okay, all right. My apologies. Lots of technical difficulties. So I'm going to just speak over everything.
Okay.
So yeah, it's a shame. I had a lot of nice slides to show you all. So basically, it's a story of teamwork. For many years, due to very high winter mortality in Connecticut, from basically 2014 through 2018, we actually saw a precipitous drop in hemlock woolly adelgid and consequently a lot of recovery in the eastern hemlocks. However, we also know that hemlocks are also subject to a lot of stresses other than the insect pests. So we also had a very severe drought a few years ago, 15 to 17, which really stressed our trees. And then recently we just had another drought, a severe and a shorter drought, but it was extreme, and it was from the summer and fall of 2020, which many of you have already mentioned. Hemlock woolly adelgid, just to give you a little bit of perspective, and I'm sorry you can't see my slides, but in 1997, hemlock woolly adelgid probably occupied about a third of the range of hemlock. In Connecticut, we first got hemlock woolly adelgid reports in 1985. So we've actually had a very long experience, over 30 years of managing and feeling the effects of hemlock woolly adelgid. As of the latest map from the U.S. Forest Service in 2021, the hemlock woolly adelgid range has now spread up into Canada, into Nova Scotia. I see a new county has been detected in 2021, and it's pretty much overtaken half of the range, I'd say, of eastern hemlock. And the other thing I want to actually mention is that in Connecticut, we don't just have hemlock woolly adelgid threatening us. We also have elongate hemlock scale. And these 2 warm winters that we've just come through, which would be 2020 and 2021, we had effectively less than 10% or 0 winter mortality of hemlock woolly adelgid. Hemlock woolly adelgid has always been, in our neck of the woods, has been subject to severe winters, and that has actually played a role in having these fluctuating populations of hemlock woolly adelgid affecting our trees in subsequent seasons. So when I do my sampling, I've been using a new technique that I found very useful. It's called blowdown sampling or wind down sampling, and I go out multiple times, especially after some extreme wind event. And all these samples are right on the forest floor. I just pick up a bunch of hemlock samples, hemlock tip samples, and I take them back and I rate them. And you'll be amazed at how much information you can get from these samples. And they're totally random, and really gives you a really good perspective, because in many of our forests, you can't really reach up into the higher canopy. And it would take too much time to do pole pruning, etc. So what I did find in 2021 was that in all my sampling, I sampled over 15 sites, at least twice. Most of the sites we had HWA in roughly about 40% of those samples. And I usually collect over 30 samples, wind down samples per site. While elongate hemlock scale was in 100% of all the sites, showing continued urgent spread of elongate hemlock scale. Now, the outcome of those serious stresses from the droughts, the 2 droughts that we've just had in the last 5 years, was that I also saw increased hemlock borer outbreaks, especially on ridge tops. But once again, we were saved because we had 2 extreme precipitation years. 2018 followed the extreme drought that we had. And then in 2021, we got increased rainfall. And that was really, I think, significant, because I'm starting to see some recovery in the sites. Once again, I apologize. I had some nice slides to show you the recovery, but I can't show it to you. But what I did want to mention is that some of this recovery does occur in areas which have not been treated for either hemlock woolly adelgid nor elongate hemlock scale. And I think if Andy Hubbard is on board here, he will remember we took a look at the Colebrook River Lake Dam, and I've been taking pictures of the hemlocks on this very steep side of the hill above the Hogback Dam, and it shows a sequence of trees being close to death, would probably have been considered dead in 2017 from the drought, recovering slowly until they're fully refoliated in July 2021. And I guess what I'm trying to bring across here is that I think that hemlocks actually can recover in certain circumstances, and that circumstance may be linked indeed to increased precipitation, which I think is somewhat hopeful, because all the climate projections are that we are going to see more and more wet years. And it may be that these increased precipitation events will occur in spring, which will be useful for the hemlocks to fuel their bud break and perhaps even put out a second flush. So that's kind of the context of it. I think that although we do have periodic droughts and they're very, very concerning and they stress the trees, hopefully we'll also get into, hopefully these droughts will be punctuated by abnormal precipitation, which will also try to help the trees recover. So just to mention again, so we did not have effectively any winter mortality 2020 and 2021, which resulted in a lot of resurgence of hemlock woolly adelgid and also a lot of reinvasions. I've been seeing hemlock woolly adelgid infestations at very high elevations in Canaan Mountain, Mount Riga, and other very cold areas, which normally HWA has not been able to gain a foothold. Well, in 2021, late 2021 in the fall, I did a lot of work scouting Great Mountain Forest, and we were alarmed to find that hemlock woolly adelgid had penetrated far into the wilderness, and a lot of it was concentrated around the lakes and ponds, not only in Great Mountain Forest, but also residential private lake communities in Norfolk. So armed with this, I then progressed to the point where I'm not able to rear beetles anymore. So from 1995 to 2007, we had a team of us working here at the Valley Lab in Windsor, and we reared and released over 176,000 lady beetles from Japan. Now this lady beetle is Sasagi sugi. It was first brought in and studied by Mark McClaw and myself, and then we developed a mass rearing method, and through our experimental releases, we released over 176,000 beetles on 26 sites. Majority were on state lands. And once again, I'm so sorry I can't show you this, but I mean the sites that we released these beetles now some 22 years ago are still thriving. They will occasionally get hemlock woolly adelgid, but then the trees are still there, full crowns, and unfortunately some of them now have elongate hemlock scale, but largely to speak, most of these trees are doing pretty good, and I'd say at least 70 to 80% survival of mature hemlocks in these various sites. So looking at the eastern hemlock distribution in Connecticut, it does look like actually, I looked at the Connecticut FIA stats from the last publication they have available, which is 2019. And it was actually very surprising because it indicated in their tables that the majority of eastern hemlock and eastern hemlock white pine forests in Connecticut are on private timberland. So really all these biological control releases that we did in the first decade or so of our research, we were not targeting private land. And also I was seeing a lot of these HWA outbreaks on private lands, especially in Northwest Connecticut. So it led to a concerted effort to expand what I think is a biological control strategy to the privately owned hemlocks and beautiful pristine areas in Great Mountain Forest and all parts of Northwest Connecticut, and also to extend hemlock woolly adelgid biocontrol on state lands. And the reason we've been able to do this is because I have partnered with Tree Savers. Tree Savers is the only commercial company that rears Sasagi skinless sugi for the public. They're a tremendous operation. The director is Jamie Bonowitz, and I've known Jamie for over 10 years. I'm sort of a technical advisor to their rearing operations, seeing as I've been working with this species for 27 years. And we've developed a close friendship as well as a close partnership. Now, many of you will know that there's a lot of other biological control research that's going on with other candidate species, such as the silver flies, and there's also a Laricobius beetle that's being released as well in other states. But in Connecticut, we've concentrated on Sasagi skinless sugi. The reason being that I'm seeing the results, the positive results in our various historical biological control sites. Sasagi skinless sugi is, I think, really pristinely adapted to the climate change that we're coming through right now, because it has 2 to 3 generations of beetles that are produced. The overwintering beetles will produce eggs in the spring, and then their progeny will produce eggs again, and then their progeny may be able to squeeze in a 3rd generation. And these overlapping generations of predators will attack all stages of hemlock woolly adelgid from spring through to fall. So it has a very long season of impact. Most interestingly, I wanted to address the ability of Sausage skimnus to withstand this very changing climate. So in 2020, November 2020, I caged a few beetles onto a low branch of a mature hemlock tree in about a meter square net cage. I caged them in early November. There were 10 beetles, and 5 females, 5 males. And I left them there, and I had a data logger on the tree with the lead inside the sleeve cage, so I could monitor the temperatures within the tree. Well, that January of 2021, we did not have extreme cold as a polar vortex, but we did have extreme wind chills. And I don't know if people remember this. At the end of January, we had extreme wind chills of -20 to -30 degrees Fahrenheit. And those poor little beetles were caged right onto the foliage of the tree, no access to overwintering sites on the bark or anywhere else in the forest. Anyway, left them in there until August 1st. The reason I left it so long was because we had such a lot of rain in June and July, and in order to try to retrieve the beetles, I lay out a shower curtain underneath the branch, and then I take off the branch, and then I tap the enclosed foliage to see how many beetles I could recover. And I could not do this because at every stage that I wanted to do something, I had such rain. But it turned out that it actually proved something really important. The beetles not only survived the extreme wind chills in January, they survived heavy snow. I had a minimum of 0 degrees Fahrenheit, about -18 Celsius in the cage. And then they reproduced during the heavy rains. I think there was one day, there was one section in June, one period in June where it was continuously raining for 10 days. I had about 7-8 inches of rain in that time. And so I have no doubt that the beetles can survive these heavy rains, high humidity, maximal temperatures within the forest in the cage was 89 Fahrenheit. So this is encouraging. I think it is a beetle that could do well in this changing climate. So I reached out to a lot of people, some of my partners. Of course, my long-standing partners are the members of Deep Forestry. But I also reached out to MDC, Andy Hubbard. I also reached out to towns because there's a lot of beautiful hemlock conservation areas that are owned by towns. It's open space, and they were very concerned. I mean, in fact, I was approached by the town of Woodbury to help them with Nonawuk Falls, which is a beautiful hemlock ravine in Woodbury, which was covered in adelgid. So I've been reaching out to all these folks, also to private forests such as Great Mountain Forest, and several land trusts have come on board. The Wyndham Land Trust is a very large, has many properties in Northeast Connecticut, and working with Andy there at the Wyndham Land Trust and with volunteers, we were able to do a lot of releases in those areas. So the system works like this. I try to help train folks on how to identify the best trees for putting the beetles on. And the main thing is that we don't do mass releases. We do releases as much as each organization can afford. So it's economical. And the main thing is timing. You want to get the beetles in when the infestation is new and the trees are relatively healthy, because the beetles respond in their fecundity and their reproduction to the most healthy infestations. It's not really advisable to release into a declining stand where there's no adelgid, obviously. So by doing this, we actually release so many more beetles. My last total I told you was 176,000, 2007. by working closely with tree savers who have actually donated over, let's see how many have they donated. There's over 12,000 beetles that they've donated to my program here. So in 2017, they donated 2,000 beetles. They were surplus beetles. It's the end of their season. They had finished all their releases to their customers. And then in 2020, they also donated over 4,000 beetles to me to distribute, and most of them went onto state lands. And then last year was their highest. They donated over 6,000 beetles and was able to spread them between state lands and also some of the land trusts and participating folks. So I really think this is a wonderful way that we can do something about the encroaching adelgid. So the latest thing that I want to report also is that I've been really supported by the Farmington River Coordinating Committee. Many of you might know that the Upper Farmington River is a very scenic and beautiful river, and it is the state's only national wild and scenic river. Well, when I was doing my scouting there early in 2021, during the winter of 2021, I discovered huge areas of fresh new adelgid invasions on all the Upper Farmington forest watersheds. So it really alarmed me. And then thanks to somebody from the Land Trust who suggested I contact the Farmington River Committee, Coordinating Committee, to see if they would fund some biocontrol efforts. My efforts were successful, and they're not only funded last year, they're going to fund again this year. And my assessments of hemlock woolly adelgid mortality has shown me that actually survival is a lot higher along rivers and around lakes. So up on Canaan Mountain, the 2022 winter was quite severe, not devastatingly severe, but severe, and they had probably -9, -8 sub-zero temperature. And the survival of hemlock woolly adelgid up there was variable. Some samples were 60%, some samples were 100%. So an overall average was about 80%. So there's still going to be adelgid up there on top of Great Mountain Forest, along the Housatonic State Forest. And I think it's just the prime time to strike and do the releases, because the beetles will find these lower densities and they'll be able to establish on the HWA there. So I'm also working with the MDC, Andy Hubbard, and I have done some releases along the feeder brooks that enter the Buckhamsted Reservoir. Very important. I mean, in Connecticut, most of our hemlocks line streams and lakes and ponds, and we really do not want to be, well, we can't do chemical control there. Now, you will hear of chemical control being advocated in other states, and that is obviously their choice to do that. But let me tell you from my experience with the public, and I work extensively with the public, all these entities do not want to use chemicals near sensitive wetlands and riparian areas. At most, I will suggest if they have a screen or a hedge or something that's a hedge of trees that's important for privacy, I will recommend they use horticultural oil to try and reduce the hemlock woolly adelgid impacts. But you really cannot use horticultural oil near water either, for obvious reasons. So to me, hemlock woolly adelgid biocontrol is the safest and the most effective way to get some management tool into the situation. And the one thing that I do want to emphasize is that the reason why Sausage skimnus is so effective is because it has such a high fecundity. So I'm just going to illustrate this by telling you how Tree Savers does it. Tree Savers produces thousands and thousands of beetles every season from January to June. And they have to collect hemlock woolly adelgid to feed and rear the beetles. At the end of the season, they store 3,000 of their youngest beetles so that they can start the whole cycle again, usually in December or January of the following year, and produce the beetles for distribution and purchase. Last year, because this phenomenon of HWA was so widespread throughout almost every state that was affected because of the mild winters. They were able to do their highest production. So from 3,000 stock beetles, which would be roughly 1,500 females, they produce 125,000 beetles for distribution. And I think that tells you alone that if we could even get a fraction of that predation power into our forests where the Dalgid is, we stand a good chance to manage HWA levels and reduce the combined stress of elongate hemlock scale, drought, hemlock borer, all that stuff on our trees. And so I just want to quickly rattle off the acknowledgements. I want to thank, of course, Connecticut Deep Forestry Division, Connecticut Deep State Parks, Steep Rock Association, the MDC Metropolitan District Commission, Great Mountain Forest, Roxbury Land Trust, Wyndham Land Trust, Southbury Land Trust, New Hartford Land Trust, Towns of Monroe, Woodbury, and New Hartford, Woodbury Conservation Commission, the Ragged Mountain Foundation, Pomfret Audubon, and especially thank the Friends of American Legion and People's State Forest. This is FALS, because they have been so wonderful in helping me to do the releases at the People's State Forest and American Legion State Forest. And a very, very special thanks to Jamie Bonowitz and the team at Tree Savers. And that's what I got, Vicki, without my slides.
Thanks, Carol. That was great. Good summary of all the work that you've done over the years. There's a couple of questions in the chat. Dave Irvin wants to know, did we ever reach what you would consider limiting temperatures in January to kill HWA?
So David, we did reach some limiting temperatures in the northwest corner. That's Division 1, Climate Division. Now, my previous research established that we had to reach -11 Fahrenheit in order to get 90% kill in Division 1. And in Division 2, which is the central area of Connecticut from northeast all the way down, including Danbury, we'd had to have had -8.3 degrees Fahrenheit. And along the coast, we needed to have -6 Fahrenheit in order to get 90% hemlock woolly adelgid mortality. It looks like we reached somewhat near killing temperatures in the northwest corner, but certainly not, as I mentioned before, I did these assessments on Canaan Mountain, and it surprised me that there was such variability, obviously showing that some HWA is surviving, and 60% is well more than enough to reinfest areas in the forest. So hope that answers your question.
Yeah, and Bob Mara pointed out that the 8 Mile River and its watershed is also part of the National Wild and Scenic River System, and I think that is true.
Okay.
I thought that there was more than one in Connecticut, but I couldn't remember which ones they were.
I think they're under application. I'm not sure whether they actually got designated. They're applying.
Yeah.
I believe.
Dan Evans wants to know, how can DEEP Forestry help continue the release work on state lands? More beetle purchases needed for state lands in 2022. That's good if we can get more beetles out there.
If we can get any timber sale money like we did back in 2010, that would be wonderful. I've just put in a permit to amend the permit that I have from DEEP, and the 4 areas that I've managed to see so far that have how should I say it, serious infestations of hemlock woolly adelgid include Kettletown State Park. Now, Kettletown was a release site back in 2006, and actually it's really interesting. The lower Kettletown area, which is near Lake Zor, is really healthy, but some adelgid is starting to pop up. When you go up on the trails, and the upper trails on the ridge, which is what the folks from the Land Trust told me to do, I've been seeing much more adelgid up there. And I would like, if I can, if Jamie is able to contribute beetles, I'd like to concentrate some releases there, because it's a wonderful system. It's further south the beetles should do very well there. We've released to the south in Wet Mountain Park, and the tree warden there tells me they've been doing really well ever since, the trees there. And then to the slightly to the north along the Pomperock River, we're now putting releases through Woodbury, town of Woodbury. Also, Flanders Land Trust is planning to do a lot of releases, and also Southbury Land Trust. So it would be a wonderful chain if we could establish the beetles along this river system. Another area that I've seen today that's very concerning, actually, and I'll bring that up quickly if I have time, is the Sages Ravine area. So when I did some assessments back in October, I went on top of Mount Riga Road. This is the extreme northwest corner between New York, Connecticut, and Mass. It's extensive hemlocks, particularly on the Massachusetts and New York sites, actually, not so much in Connecticut, but there is a beautiful ravine there on the way up there. I was very alarmed because these sites represent my baseline sites, and my baseline sites are how I measure the recovery in adelgid infested sites with sites that are at high elevation in Connecticut, which have really never had any pest infestations. And what I found was the first time that I've ever seen it was along a trail at the Appalachian Mountain Club cabin. They own about 250 acres at the top of Sages Ravine. And I wandered along the trail there and I found significant HWA just at the beginning of this trail. And it really surprised me. It was not in my baseline site. This is beyond. Since then, the folks at AMC, I alerted them all, and they went up and they said it's through an old growth stand that they have up there. And I've also been contacted by folks in Mount Washington, which is the town across the border, and they are terribly concerned because they said it's all over that mountain. It's all over Sheffield, Mass. And Sheffield, Mass abuts Salisbury, Connecticut. So the other area I would like to address, if we get some beetles, is the Mount Riga Scenic Preserve. This is the bottom end of Sages Ravine, right on it's in the town of Salisbury. It's state land, and I was out there last week, and I found a knoll of hemlocks that had some active HWA. And then another area I'd like to try to address, or at least scout out to find some good spots, would be the Housatonic State Forest. And the Housatonic State Forest adjoining Great Mountain Forest and all those, the Canaan Mountain Scenic Preserve. I think that would be a real shame if we have a decline occur up there. So it would be nice to put some beetles there. So sorry for that long rattling thing, but if you have any other areas that you're seeing a resurgence with HWA, please let me know.
Sounds good, Carol. Thanks to everyone. I just want to mention real quick, I had a question in the chat concerning brown-tail moth. Someone wanted to know how concerned we are about brown-tail moth, and I will say we are very concerned because brown-tail moth is not only a defoliator, but it also can be a pretty significant hazard to people. Brown-tail moth is one of those that has irritating hairs when they're caterpillars, and the hairs remain irritating for 2 or 3 years after the caterpillars have gone away. And this irritation can range from a pretty severe rash to a full-on allergic reaction. So not only is this thing nasty on the trees, but it can be nasty to those of us that are out walking around in the forest. So if anybody else has any questions or comments, now's the time. Like I said, this whole thing has been recorded, and I will post a link to the recording both on the Experiment Station website, and I will email it to everyone on my list of participants. And of course, you can share that with whoever you think might be interested. Mark your calendar now. The Forest Health Monitoring Workshop for 2023 is scheduled for March the 7th, and I believe that's a Tuesday, so March 7th, 2023, here at the Experiment Station. If no one else has any other questions or comments, I'm going to sign us off. Jeremy Clark just commented to Carol, I'm more than happy to work with you on potential release on Housatonic State Forest. He's going to send you an
Thank you.
email.
Thank you, Jeremy.
So anyway, for the good of the order, does anyone have anything else? If not, thanks to everyone who gave presentations today. As usual, it was a very informative, very rich presentations, and thanks to everyone for sticking in as long as you have.
Thank you, Vicky.
If there's nothing else, we'll stop. I'll stop the recording and we can all go have lunch. Thanks, everyone.
Thank you, Vicky.
Bye.
Thank you.
Bye. Thanks, everyone. Stay